Hi, Danny.
Hey there, Blint. How you doing?
I'm doing all right. How are you, sir?
Doing really well. It's good to see you. It's good
Good
to
to
see
see
you.
you too. How was your day today? Good science?
Good. It's been good stream today. Good discussion. Man, it's been busy this week though. I've got museum projects and research projects, and I've got an abstract that I need to get submitted soon, and oh boy, there's a lot going on. But and we had that meeting this morning too. I don't know how much we should talk about that, but exciting things going on. Exciting things going on.
Yeah, I'm not sure what we can say yet. It was kind of a mystery, but
Uh,
yeah, there are things in the works, y'all.
yeah, I'm trying to do our best to kind of grow the science community here on Twitch. There are exciting things happening. I'll say that.
Yeah, yeah. It's and Danny, y'all, is the best one to lead the charge, as it were.
I don't know about that.
I will tell you. I will say it. Magistrate,
I
thank you for the
appreciate your
biddies.
confidence, Blint. Yeah.
um,
Uh,
so where would you like to start today, Danny?
um, maybe with well, shoot, I was gonna say with the paper that involves some of the people I used to work with, but there's multiple papers from you and from me this week with people I used to work with. So I don't know. What do you think? Do you have a preference?
I don't. I think they all go like they're all a very similar theme, right? Of where we can
Um,
save and get genetic information from, and
let's
so
do the one with I think it's Bayul et al. Alita Bayul's paper, the review paper of ancient DNA. I think that's the one that I'm most excited to talk about, I guess. And if we can only get to one, I want it to be that one. So let's start with that one.
yes, awesome. So I have it
That being
pulled up.
said, I have until, let's say, let's see, 5:30 here. Let's say I have until 8 p.m. today, so 2.5 hours.
Okay, sounds good. So we'll get through
Yeah.
half the paper.
Yeah, we'll see.
And I mean that in the best of ways, y'all. We just kind of start rabbit holing. We get really excited about the science we're chatting about. So it's, it could be a little hard to stay on task, as it were. But I think that's the point, right? It's like highlighting that we can get excited about different kinds of sciences, and we can, come up with new ideas and come up with like the next steps to really analyze.
Absolutely. Yeah, you never like I don't know. You as a geneticist and systems biologist, molecular biologist, you're gonna have insights that I never would have thought of, and maybe I'll have some semblance of an insight here or there as well.
That's why
And
I'm excited for these papers, is because I feel like
yeah,
these really hone together both of our unique skill sets, and kind of like even in some of the like extra parts of these papers where they're like, oh, these will be a really nice extra something to have together. Suitable substrate, thank you for the follow or the resub even for a year.
Suitable Substrate just subscribed for 12 months.
Thank you very much, Suitable Substrate. That's a piece of raw science and another partner plus point. Welcome to Hack In. Thank you so much for that. Yeah, so like in the paper, what was really cool is that there was this whole perspective part that they were chatting about, that wouldn't it be great to have something along the lines of like molecular biologists and paleontologists working together to really hone in on these questions. And I'm like, that kind of hits at what we do here on the stream every day.
I guess so. Yeah, yeah. It's neat. I guess that's what E.O. Wilson would call consilience, right? Where you've got these different fields that can kind of mesh together in order to provide insights that wouldn't otherwise be possible.
Yeah, that's what
Yeah.
I'm really looking forward to. Danny, do you by chance have a little boost you can do on your side of the volume? I don't think anything's changed.
Try,
It
yeah.
usually doesn't.
Everything's the same on my end, but I can try and turn up this dial here.
Yeah, everyone's saying that it's.
Let me put a little mark on here so I know where the old spot was.
Sorry about that, Danny.
It's okay.
Thank y'all for the hype train, by the way. Really appreciate you guys. For all those resubs coming in, those biddies. Thank y'all very much.
Is that better?
Is that better, y'all?
Testing. Yeah, it's weird. Everything might be exactly identical to what it was last week, and yet with, I don't know, the particulars of Twitch guest star, stuff just goes askew in weird ways. We have no way of really telling what's going on.
Maybe a little more, it seems like, from the chat. Because I have you boosted all the way.
How's that? Is that better?
Definitely sounds better on my end. What do y'all think? That's definitely louder. Yes? Okay, we got a bunch of yeses. Awesome, cool. Good to know. Hi, Kimmy.
In 10 minutes, I'm going to be deafeningly loud without having touched anything.
No, we'll have changed.
Who knows what Twitch guest star is doing behind the scenes?
Nothing will have changed at all. And like earlier this week, Danny, we actually had a really exciting stream on Monday. We did a collab with a professor from Laurentian University, and he put out a paper on isolating DNA with at-home goods. So using a coffee filter, isopropanol, water, a Ziploc bag, a rubber band, and like a banana or your spit, you could isolate DNA. And so he and I did both kinds of isolations together, and we had people in chat who were also doing the isolations from home and posted pictures on the Discord with their isolated DNA. And it was like,
Very cool.
He was on guest star, and the audio was, at that point, fine. Nothing's changed, but it was really cool to have people doing experiments at home and on stream, like from the learning process. And it was a really fun one to be able to do, yeah.
That's awesome.
It was I thought a very unique way of going about it. So just wanted to be like, oh yeah, we did that thing. I'm like, that's cool.
That's pretty darn cool. That is pretty darn cool. I've been thinking about maybe doing something similar with like trying to teach basic cladistics. So, creating a cladogram, like an evolutionary, a model of an evolutionary family tree. But it might be a little bit more involved than that, because people would have to download software. And,
I wonder if
it would be quite as hands-on and fun, but it would be instructive, I think.
Definitely. Yeah. I wonder if it's something like if you made a PDF ahead of time, where it's like almost like categorizing certain features, like so maybe not starting off with like a real life example, but like just like we make up some features and we build the tree along the way. That might be an easily accessible starting point.
That's kind of the idea, but the software aspect of it is a little hairy. That's what I had trouble with when I did this back in undergrad. So I don't want to make it too difficult. But I should see if it's gotten easier nowadays, if there's anything that's a little bit more user-friendly.
That's the hard part. If it's a free software, then it's not user-friendly, and then if it is user-friendly, it's a $5,000 paywall or something like that.
Yep. That's the world we live in.
And speaking of paywalls, y'all, these papers that I picked today unfortunately did have paywalls behind them.
But I appreciate you sending the PDF.
Oh, yeah.
Yeah.
And the PDFs are uploaded on the Discord, y'all, if anyone's interested in getting the papers. I will post a link into the chat, but that will be blocked, unfortunately. Luckily, my old university access still came through. But yeah, where I thought we kind of leaped off this last week was we were chatting about DNA in cells from dinosaurs, and I don't even know how that rabbit hole came up, Danny. But we were just kind of talking about, well, that would be really cool, and I wonder what exactly is going on. And I mentioned, well, there were some papers that looked at and claimed to have gotten cells with DNA in them. And I think that we're just like, all right, let's go ahead and just chat a little bit about those and see like what the state of research is on them. And so what I was looking in the literature is anything really new. And this isn't the newest paper. It's 2021. I couldn't find anything that was really up to date with like a brand new release where they were actually looking at DNA and trying to capture what is called aDNA, which isn't a term I was familiar with, but it's called aDNA for ancient DNA. Which I guess, it's makes sense in the names of the terminology, but I've never heard of it like referred to it as that.
I honestly hadn't either. No, but I've seen it in some other papers since then.
Yeah.
In looking more into this today, it does seem to be a commonly used term.
It was in your paper too, with the looking at the proteomics component of it. So then I was like, okay, this is nice that it all kind of connects together. It makes sense.
Yeah.
Like I've heard of eDNA, which is environmental,
Environmental DNA.
yeah, where you can sample the air and like identify what animals have like come through that area, which is really cool in terms of tech too.
Or the water. That's one of the reasons why it doesn't really seem like there is a Loch Ness monster, because if there were, you'd be getting eDNA from it. And if you're getting DNA from elk and weasels and critters like that from around the Loch Ness Basin, you're getting their DNA in the lake, but you're not getting the DNA from some sort of gigantic creature that's supposed to be living there, then chances are it's just not there.
Yeah, which is... you were talking about that on stream last week, right? Like dispelling the notion of the Loch Ness monster for that reason, and I never really thought of using eDNA to dispel that kind of rumor, but it's a really cool way of doing it, right? Like it is. Yeah, here's the evidence.
Yeah. Way of like testing that, because it's so difficult to prove a negative, but it's with eDNA you get pretty close to doing that. So yeah,
it's a really impressive tech. Yeah, and Sloppy Salamander's wondering if there's a difference between DNA and aDNA. So it's age, honestly, is the big thing, and that it's a sliding scale and squishy term because it seems like different papers categorize what ancient DNA is differently in terms of timeline. So is it going to be a 2,000-year-old cutoff, a 20,000-year-old cutoff, a 65 million year old cutoff? It kind of just all kind of mushes together, Sloppy Salamander. So that also makes it a messy sliding scale because the amount of DNA degradation that you'll have across time is also going to vary. So I think more of it's this all-encompassing term of this is old stuff that is, I think, probably older than 100 years is honestly the range that I was seeing, which is a little bit frustrating that it's such a big... It's like, yeah, I guess Dame Karen's right. It's like the retro game category, where Crash Bandicoot falls into retro, but so does like old arcade games from the 80s and everything in between. I like that analogy. Nice, yeah. And then, Danny, do you think eDNA can be used to dispel Bigfoot as well?
Oh, yeah. Yeah, well, I don't think we need it for that. I think it's pretty well dispelled to start with. But yeah, yeah.
I was disappointed that the that famous photo of the guy going like this is actually a person in an outfit.
Yeah, yeah, and we even like know the name of the guy and everything. I think his name was Bob Hieronymus. There's a really interesting book about this. But anyway, yeah, the Patterson Gimlin footage. You know, yeah, don't get me started.
But where we can get started, I think, is this really cool paper, y'all, and it's kind of a summary of the field so far and different types of dino and ancient DNA collection methodologies. And I think it's exciting because it argues a lot of unique features of DNA that maybe we haven't considered before. Now, it doesn't mean we're going to be able to extract dinosaur DNA, y'all. Like, that is something that the paper was saying that at this stage they are not able to do that. But what it was trying to dispel was, can we even visualize DNA in these cells? And so there's a bunch of these unique samples that they were looking at in the study from different eras, from different organisms, right? Everything from plants to dinosaurs, and a lot of cartilaginous cells when it came to dinosaurs. And they did DNA stains that we do in the lab every day. So they did propidium iodide, which stains nucleic acids. They did DAPI, which binds to DNA in particular. They did a host stain, which is a similar flavor to DAPI. Essentially, it's just binding to nucleotides. And so that's what you're seeing here, is that these positive, like, white dots here, or the red mass here, or like this one, which I think is really clean, a 55 million year old piece of fruit, those dark circles are the DNA within the cells.
That's beautiful.
Yeah, and this one honestly looks like an image that you could have told me it was from today.
Uh-huh.
And I would be like, yeah, sure, because it looks really, really clean. What they are pointing out is that while they're able to get these, that doesn't mean that the nucleotides in the DNA are any kind of collectible state. They're just kind of in a broken down state, and we are able to get to them in terms of staining, but they made it a big point in the paper, y'all, that this isn't extraction. They're not able to take this out, and if they were to start that, it would be very, very different than just doing this kind of stain, which I thought was very interesting, that there is this difference of just doing a stain versus actually prepping the sample, removing the minerals, removing any kind of contaminants, and then actually getting anything out.
So I was actually hoping, Balint, that you could further outline some of these terms. When we're talking about extracting DNA versus sequencing DNA versus staining DNA, what do each of these mean, and kind of like at what levels are each of these in terms of complexity or difficulty?
Yeah, so I think what's really cool is that in the methods section they went over how they wore these insanely like complicated and sterile suits. And actually, Danny, they mentioned tech that you had mentioned to us on your field visits and at museum sites, right? With like where they have those clean rooms where you're extracting the dinosaurs and there's all those vents coming up, right? And you're getting stuff out from those vents.
The dust collection,
Yeah, like they had that on top of bleaching everything down to prevent external DNA from coming in. And actually, for returning chatter, the British Asura, DNA from amber is very difficult for a couple of reasons, primarily because of contaminants. So you could be able to get some kind of stuff. They even mentioned it in this study that having things from amber is going to be highly contaminated, primarily because you're getting in all that plant matter, bacteria, and of course the resin itself is full of antimicrobials, like from the tree itself. So it, like they pointed out that while it preserves like an insect or dinosaur skin, even whole birds sometimes, there's going to be a lot of contamination in that, even like independent of the scientists doing anything with it. Built into it, there's a lot of contaminants, and so it's a very difficult thing to pull out. But it's a really good question. But you're not going to be getting dinosaur DNA or even insect DNA. Like you're just going to be getting probably plant matter mixed in with a bunch of different things out. Yeah. So Danny, with your question, I think that's really good. There's like the way I was going about it was there's this clean room that they have, and so everything that we're about to mention has to be under the guise of it is really hard to do this from a cleanliness standpoint, that you are not contaminating things.
Yeah.
And it's not just this paper, y'all. Like Danny's paper that he was highlighting today too of the proteome is the exact same thing, because there is DNA on your hands, there is RNA on your hands, there are proteins on your hands. If you are not clean or your equipment is not clean, that's game over, right? Like in terms of
contaminate everything. Because if you're looking for just the tiniest scraps of DNA, in these fossils, you got to make sure that you don't have anything on you that might mess up the sample, because you might accidentally mistake some modern DNA for ancient DNA if you contaminate that, and then, like you said, it's game over.
Yeah, and it's the same with doing these stains as well. So we had a question from, I think, Cass. Could the stain be reacting to something other than DNA? So Cass, these stains that are done here, these propydium iodide, this DAPI, is done every day on living cells and non-living cells. And it always binds into the helical backbone of DNA. So in terms of the chemistry, that's where it particularly inserts itself. And so does it insert itself, let's say, into a protein, right? Which would be very common in a cell. No, it doesn't. It only binds towards nucleic acids. And actually, you can make it very particular. So propydium iodide does single strands, so like RNA and DNA. DAPI just does double-stranded, which is DNA. And so you have that level of distinction. Now here, this image here that they did with the propydium iodide state, I'm not about to suggest that they pulled out RNA. And neither do the researchers, right? Like, you sneeze on RNA and it is gone. Because there is things in the air on your skin called RNase. And RNase is an enzyme that breaks down RNA. There's also DNase on your skin that breaks down DNA. Proteinase that breaks down protein. So it's always the ace on the ending.
There's a pattern here.
Yeah. It's the lovely Latin roots, right?
Yeah.
And so there's ways to... there's this chemical called RNase zap that you can spray your body down with and all your equipment, and that gets rid of the RNases that when you're working at a lab bench. But it lasts about 20 minutes before the air in the surroundings has like now gotten enough of a density back on your equipment where it's going to be a thing. So, you know, as Sam asks, how do you clean your hands and getting ready for this kind of protocol? You have a clean room. And all your equipment is, I think they bleached it down with some absurdly high level of bleach plus hydrogen peroxide. And so it's like really nasty chemicals because you're trying to rid every ounce of genetic information that's free floating. Danny mentioned, right, environmental DNA, where you can measure from the environment the animals that are like just in the area, right? You can do that in a lab on a person scale. Right, and so it's like, how do you avoid doing something like that? You have to be extremely clean. And so it's like the prep work going into these experiments is very much on that point. And I think what was nice was the paper pointed these things out. That, because, right, immediate skepticism would be, oh, this is just contamination. But I think what's neat is that these are multiple different ways of replicating the same experiment on different samples, and they all have these especially clean rooms. Which is key to have. Hi Skip, welcome in. I worked in an RNA lab, says Forrest. A few years last millennium, it was maddening. Yeah. Forrest, I found out, so in my work on leafcutter ants, that leafcutter ants have a very particular kind of RNase that is more active than transient RNases. And so the protocol for getting rid of them is not sufficient. And from other ant species or flies, like, it does not work. And so we had to adapt things, and it took 6 months to adapt just to extract RNA that wasn't broken down.
Holy cow.
Yeah, and so you're like,
The
well...
results were worth it in the end.
well, so we were able to do the sequencing and figure out, like, the RNA is different between these casts. For, like, these are the genes that are different between them. But without that, like, you would have had just degraded product, and you have no way of knowing what exactly is going on. So that's what something living. Imagine now this as something that's millions of years old, and you're trying to do something similar, right? It's.
Yep.
I think it's remarkable they have anything out.
It really is. It's astonishing that we've got any traces at all of any genetic material from animals that are tens of millions of years old that died that long ago and have been buried in rock for that long. It's nuts that we've got anything at all.
Yeah. And so this, to get back to your question, Dan, like, how easy is something like this? So this here is, if you go by the protocol, they take these stains, which is in a vial. So imagine you have a tube full of this liquid called DAPI. 2 Jeffs, 1 cup. Thank you for the follow. Welcome in. A tube, with a liquid full of DAPI. A tube with a liquid full of propydium iodide. Like, that's all it is. You dilute it down to a very, very small concentration, because it's highly, highly concentrated in the vial, and you do like a 1 to 1,000 ratio. So 1,000 microliters of your DAPI, 1 microliter of your DAPI to 1,000 of a liquid that's meant to be like a buffer solution. So like a water with a salt balance in there, right? You mix that together, you put it onto the cells, you let it sit on living cells 10 minutes, you take it off, you image, and you have an image of a nucleus. Like, it's something that, I would say anyone can do it at home, just make sure you wear gloves. Like, because what it's doing is it's inserting, these molecules insert itself into the DNA. And so you can imagine if you get it on your hands, like it inserts in living cells, right? And so you could be inserting things.
You could be causing mutations in your own cells.
Yeah, like they are carcinogens. They can be cancer-causing agents, right? So it's not that you at home can't order this and do this very easy experiment on living cells. You absolutely can. You just want to be very careful when doing it. It's similar side to a saline solution. It's buffered for your organism of choice. So that's one of the difficulties in this experiment too, is your very beginning experiments up here, that's essentially what they did. They took the solution, they put it on the cells, they took some cuts of the bone, and they found it was binding to DNA and it was lighting up like this. The later experiments that I think look much cleaner, for example, the plant, or one of my favorites here is this in panel F, they use what is called Triton X. And so Triton X is it's a soap solution, basically, with the fancy name. And it permeates the cell membrane. So it puts little tiny holes in the membrane of each cell so that the dye can go easier into the cell and can be washed out easier. Because what you see in the one above me, like, I guess that's a nucleus, but it's very, very full. And it's not very concentrated in terms of, or it's overly concentrated in terms of signal. And because things get stuck inside the cell, you can't really get them in and out. But instead of this, you can wash it with this Triton, so you put these little tiny holes in the cell, and then not enough to let the DNA out, but that the dye can go in and out. And so, you put the dye on, you let it sit, you wash it off a few times, and then you get this much nicer image where you can see the cell, and then you can see a very strong point within the cell, and that's the nucleus there. So those, they're the more straightforward experiments. I think the hardest part of this is the pre-sample processing, and that, Danny, you can speak to us about that, of keeping things clean, preserved when you're doing the actual experiment, so you're not destroying the fossil. Also just getting the samples as well, and telling researchers, because you were telling us about when it's the original sample, how hard it is, right, to get a hold of it, just to cut a little look in to see the age of the specimen with the haplotype. So imagine you're not going to be taking a cross section and staining them.
Yeah, I mean, well, yeah. And I also want to emphasize too how this sort of thing is very atypical for paleontology, where this idea of having samples that are clean and pristine like this, that's not normally the sort of thing that we would be doing in the field. Oftentimes, these fossils that we're finding, we're trying to get them out of the ground by hook or by crook. We're doing it carefully, but in the past, well, I'll put it this way. There was that 60 Minutes piece that I like to show a lot about B-Rex and Mary Schweitzer and her work with this stuff. A lot of Mary Schweitzer's work was cited in some of these papers. But the narrator says, paleontologists didn't used to worry about getting, sweat or glue or even the occasional spilled beer on some of these fossils. And that's pretty true. Up until the last couple of decades, we weren't really collecting fossils to be able to do this kind of analysis on them. And so, yeah, this is kind of changed the way that some paleontologists work in the field, where there are paleontologists who actually go out there with surgical gloves nowadays to collect fossils just to make sure that they're not getting any of their own cells or anything like that, on these specimens. I've still never worn surgical gloves in the field to collect fossils, but maybe someday, if these methods become more commonplace, then that could happen. That's one of I think the exciting things about this paper. It's essentially a review paper, and I don't know if you saw this, Bullet, but there was a paper that came out a little while ago, and it was suggesting that review papers might be the best accelerant, apart from just outright funding, for advancing a particular field. It's if somebody writes a really good review paper, that can help advance that field by leaps and bounds pretty quickly, if you're just providing a good summary of everything that's known about a particular field of research.
If you have the right person writing it, and that right level of excitement put in, that could, you're saying, it could be an absolute game changer. I've also heard, just so some behind the scenes things that people put out review papers to boost the metrics of the journal, because those are more likely to be cited.
To get cited, yeah.
Which is like, so it's not ever that straightforward of like, it's necessarily going to further us, but rather like there's this unfortunate background as where of like the extra reason that people are doing it. But yeah, I mean, I like this as a review paper.
Oh, sorry.
Octavius, thank you for the resub, my friend.
Very nice.
17 months.
Octavius just subscribed for 17 months.
Thank you, Octavius, for that resub. Another Partner Plus point. Thank you very much. And welcome the heck in, Sparky. And have a good one, Sam. Appreciate you. Sorry, Danny. Go ahead. I was just saying like, how cool I think that this review is. Like, I think this is a well done review.
Yeah. I think this is a review that hopefully will help advance the field. But speaking to your point as well, I met a researcher once, and she's like, "Yeah, I write, 4 or 5 or 6 review papers every year. Make sure I do 4 or 5 or 6 review papers every year because it, it helps my citation rate. It juices her numbers.
Yeah.
And there's some people who have kind of a mercenary view of that sort of thing. This paper did not feel like that at all.
No. I mean,
this is solid stuff.
I think it's a commentary too on like that publish or perish mentality, right? Where you have to have these metrics, and that's what people watch, and not like how good your science is. Which it sounds like that's what that mercenary attitude was like. I'm just gonna keep pushing just for the sake of pushing.
Exactly. Yeah.
But I thought it was I got me excited about this study.
Yeah, me too. Yeah, for sure.
And then you asked like what the next difficulties are. So I had kind of picked out this second paper as well to kind of get at some of those answers where it was ancient DNA preserved in fish fossils. And so
I almost feel like fossils here should be in quotation marks, but we can get to that in a bit.
100%. And so the only reason I just jumped over is because the question you had of like, okay, what would we need to do for something to be successfully sequenced? And so here it's something that's 20,000 years old, right? Not, let's say, 70 million years old that we're looking at.
Yeah.
And so there's like, take it with a huge grain of salt, chat. There's a lot of differences here between the two studies. In addition, they actually do get genetic information that they're able to sequence. Sequencing means that you're able to read the barcodes of DNA that are inside of the preserved cells. They did find DNA degradation, so the DNA had kind of started to fall apart at 20,000 years old. Right? Imagine what it is at 70 million, if 20,000 is already coming apart. And the big thing is that it's coming apart into smaller fragments. Now, that's not all bad. If we take, Danny's DNA or my DNA and we're trying to sequence us, we need to break that DNA strand into different smaller segments so we can do the sequencing. Because there's not an enzyme that exists that can bind onto, let's say, the top of Danny's chromosome 1 and make a copy of it all the way to the bottom of Danny's chromosome 1. It just like the enzyme will fall off at near the beginning because it is able to, insert for X amount of base pairs and then it just stops. So there's none that are that good. And so you have to break up the DNA. So, at a certain point it's a good thing, but here it's starting to break up to the point where it's going to be smaller and smaller, and if you make them too small, it ends up being the size of what are called your tags. And you always put the tags on the ends of your DNA strands to be able to do this sequencing information. And so if your tag is the same size as your DNA, you can't tell which is which on a gel. And that's your next step, is that after you do the sequencing and you amplify what your DNA is, you need to extract the right size molecule to really do the final level of sequencing. And it's all by size that you tell them apart. And if your DNA fragments are the same size as your tags, it won't work. And so that's one of the catches here, is that this experiment with the fish, the DNA is still long enough to be able to be utilized and sequenced and like done with those tags. But the ones on the dinosaurs that we were talking about, those are too small to be able to do. They mentioned the possibility of using these new kinds of sequencing techniques that don't need the tags, but a lot of that depends on you having a genome to compare against. So here in this fish study, they have a ton of sequenced fish that are close relatives, right? 20,000 years is relatively close, that they can compare against and fill in gaps. With the dinosaurs, I think even with the bird genomes, right, Danny, I'd argue that those aren't close enough.
No, definitely not. Definitely not.
And so that's some of the difficulty is in coming in with the sequencing question, is that there's so much that we just fundamentally can't sequence because of the nature of the DNA strands currently. It's not because we won't necessarily have the tech one day, but the current tech won't allow it. Does that answer your question? Like, what do you think, Danny, like, about that? Does that make sense?
Yeah, I think that makes a lot of sense. And I feel like that's some good background information that people can use to better understand the ideas in the papers that we're going to be discussing.
Yeah. And it's like we can see the DNA chat, and that right now is the extent of what we can do. We can't do any more than just, you know what, there's DNA in the cells. Because the molecules that we have glowing, all it does is make sure that it, like, binds to the glowing one. And oh my goodness, we just had someone in chat redeem 210,000 channel points. Holy guacamole, JD Does Dev. Coming in here for that VIP redemption. That's a lot of channel points, DD. Let me go ahead and add you. Before I forget, I don't want to forget. Let's quickly add you as a VIP, my friend. Thank you so much for 210,000 channel points. That's huge. Big congrats, JD. You now also have an additional sound alert that you can use. Let us know what you'd like, and it'll be triggerable by you. Just like Danny has exclamation point paleo, which is Indiana Jones saying it belongs in a museum. Thank you, JD, for all that support. I appreciate the heck out of you. We had a question, Danny, from Sparky. If we were to look at and utilize genomes, kind of like that fish paper, in terms of comparison, what would we most closely look at? Like, would it be a bird? Would it be a crocodilian? Like, especially at this distance.
The typical one is usually like an ostrich or an emu. They're going to have the most DNA in common with non-avian dinosaurs. Crocodiles are going to be pretty far. I see Sparky was also asking about crocodiles. Crocodiles are more distant from dinosaurs, whereas birds literally are living dinosaurs. So they're going to have more similarities in terms of most of their genome, I would imagine. Yeah.
I think a further difficulty, though, with the birds that I never realized until you taught us this, is that there's so few birds actually around. Like, they bottlenecked. You lost so much diversity. And that, to me, is always so freaking surprising. So that means, like, if we were doing birds as a comparison, what is the likelihood that the Allosaurus that you're looking at is going to be closely related to the few birds that we have around today?
Well, it's more likely than the crocodilians, because crocodilians also went through maybe not as severe a bottleneck, but there's not very many species around today. There's like 30 extant species of crocodilian, something like that. I forget the exact number. Birds, we've got a lot more, but they did go through a bottleneck, so birds they're at once more diverse and less diverse than crocodilians. And depending on how you look at it.
I guess they went through the dinosaur lineage, right? So there is inherently that common ancestor versus the crocodilians are even earlier.
Yes, but at the same time, not only did birds go through a bottleneck during the extinction event at the end of the Cretaceous, but also, as far as we know, all birds descended from one species of dinosaur.
Okay.
So they form a clade with a common ancestor, and that's one species of theropod dinosaur evolved into all of the bird species that we have today.
So I guess it's a double bottleneck.
Yeah.
In a way. And Chrissy's asking, what do we mean by bottleneck?
Basically, like, kind of like the neck of a bottle constricts and it gets narrower. You had a bunch of different bird species that existed during the Cretaceous period, and almost all of them died. So almost none of them left their descendants except for just a few. And so you've got this like narrow line of descent with like maybe a handful of bird species that survived that event and then evolved into all the birds that we have today.
So like here, I can do a quick little sketch. It's like if we've got a scenario, Chrissy, where you've got this evolutionary lineage, right? You've got a bunch of lines of a family tree, and we've got this common ancestor with this dinosaur, and from there evolved your modern day birds, and you've got birds that do behavior X, Y, Z, A, and B, and they're really, really diverse. And then we have a big extinction level event that takes out, 3 of these. You're just left with what is able to survive, and so you're having a smaller representation of the population, and therefore the features that we might have seen across the tree you won't be able to see. And so that's why I was curious in Danny's take, is if we did just general bird sequencing, will it be a narrow view of things?
Sorry, I was thinking about another question that Chrissy Carson just asked. Could you rephrase your question, Bullen?
With the dinosaurs and the birds?
That's the question that she was asking, yeah.
Yeah, so it's like having, and Charlie's Dragons has this nice graphic that I want to pull up too. So,
that's exactly the same one that I was about to put in the chat.
Perfect.
There it is. Yeah.
So I guess, like, with this evolutionary lineage, right? Like, here is this bird. Sorry. Oh, Mothra Main, thank you for the follow. Welcome.
What a great name.
It is, yeah. Very nice name, actually. Yeah, I like that. So we've got the bird up here.
Yeah.
Right? And that bird is related to, let me pull it over a little bit closer. These dinosaurs a little bit further down, and presumably there were other birds here that died off.
Definitely, a bunch. Yeah, and I should also point out to the chat, because a lot of people are unfamiliar with this idea. When we say that animals are related, we mean that they share a common ancestor. We mean that they evolved from the same ancestor as their relatives. So they're related in a literal sense. It doesn't just mean that they look similar or they've got, the same vibes or whatever. It's like, no, they actually evolved from the same ancestor as their relatives. That's why we say they're related. And so crocodiles are related to dinosaurs. They share an ancestor with dinosaurs if you go further back, but birds literally are dinosaurs. Their ancestors were dinosaurs, and so that's why we say that birds are dinosaurs and crocodiles aren't. Yeah.
I'm trying to see
if you look up there, to the right of the pterosaur balloon, you'll see the crocodilians.
Let's see. Aha, here we go. Yep.
Yeah.
So like right here, Chrissy, this is, and I'm chat picked up on it too. You were saying the same vibes. I'm like, I love that analogy. That's such a good way of saying it.
Because you'd be shocked at the number of people who have never learned about phylogeny or common ancestry in school. As intelligent or well-read or well-educated as they might be, sort of thing just doesn't really get taught in most United States schools, at least, until maybe college. So a lot of people think that, like, oh, well, different critters, we say they're related just because they kind of feel similar. It's like, no, it's actually based on their ancestors, what they evolved from. That's how we classify living things as scientists.
I guess that used to be the classic way of identifying them, right? It's just like very like not even
before we discovered evolution. That was the way that this is kind of before people called themselves scientists. The word scientist didn't really exist until like Darwin's time, I guess. But before him, you had people, natural philosophers or naturalists or even natural theologians, they call themselves sometimes. People just looking at animals and doing the best they could based on vibes, basically. Before we had an understanding of ancestry or change through time, or DNA is kind of the shortcut nowadays. Yeah.
And like not even really morphology back then, as much as it is now. Like, because you're right, you're stuck using morphology too, and like not even being able to get to the DNA, so you have to examine, and then like modern examine too, right? Like you were telling us, like the similarities between what it looks like now versus what it looked like then, and like, you know, infer function and from form, and yeah, it's definitely not vibes anymore.
Yeah. And I think that's what's so cool about molecular phylogenies, is that, like, the characters that you're coding are not arbitrary in the same way that a lot of morphological characters might be. And so yeah, I think that's really neat, and it's really cool what they were able to do with that, the cichlids in that paper. But wait, before we move away from this screen though, I just want to emphasize the point for anybody watching who might not be familiar with this, that like the critters that you see next to each other on this diagram, they're going to share a lot more DNA than critters that are on the other side of the tree from one another. So like that bird, that magpie in the upper right, and that troodontid right next to it, these guys are going to share much more DNA than the bird is going to share with, say, Spinosaurus, or with Triceratops over there on the left.
So Chrissy, like this point here, as Danny was saying, the bird and the troodontid here, that's their common ancestor. So from this individual evolved these two branches. And so if you want to see, like, who is this bird most related to, you go down the tree and you hit this common ancestor, you see, okay, the troodon is the most close relative. And then if you want to see, if the question is which is it more closely related to, this troodon or the triceratops, so to get to the triceratops' most common ancestor, you have to go down the tree here, down again, and that gets you to the start of the dinosaurs.
Yeah. And
then you have to go back up the tree to a split point to get to the triceratops. So you have a lot of common ancestors to jump through, and then even, it's not just that you get to this common ancestor where the start of the dinosaurs is, that branching takes off in a totally different direction. So they are a massive worlds apart on the tree, which is kind of cool to see, like, the difference between a triceratops and one of these magpies.
Yeah, good explanation there. Yeah, and this is at the core of biology, basically. That's why I'm so frustrated that this sort of thing doesn't get taught in most American schools, at least, because it's absolutely fundamental to our understanding of the history of life on Earth. And most people never encounter this until maybe they go to college and take a biology class.
Yeah, and I feel like it's a little, it's opportunity then, right? If you don't have the opportunity to go to college, which a lot of people don't, you won't.
Absolutely, yeah.
Like, this is almost fundamental to understanding also why things like viruses infect us, or how they can jump from certain everything comes back to this like basic understanding of relatives, right?
Yep, it's the fundamental principle in all of biology, basically, and it's nuts to think about how many otherwise extremely intelligent, well-read, well-educated, worldly people are walking around making decisions, you know, running society, and they've got zero understanding of this whatsoever. Like, the number of doctors and lawyers and, you know, senators and whoever else just have no notion of this whatsoever. So everybody in chat, I'm glad you're here. And hopefully you can help us, I don't know, I was going to say spread the word about this, but, yeah.
Change the world a little bit, right? Actually, Coffee Rock said that. Coffee Rock said too, be on Twitch and just learn here. And that's our mission, right, of making this accessible. And like, Danny is the expert. He is in the field. He does this for a living. Like,
I mean, shoot, you're also working with evolution in your field, though, Bullet. I mean, it's something that's absolutely fundamental to all of biology. Like,
it really is.
Obviously.
Yeah, no, yeah. I don't know of a field that doesn't take into account nowadays some level of phylogenetics. Because if anything, your funding is based off that.
Yeah.
Like, doing this will be helpful because it'll inform us about humans.
Yeah.
Right? Yeah. Like, basic understanding of it. Moonrise Rabbit says, Danny, I'm going back to school in the fall for an Associate of Science degree. I'm going to be helping too.
Very, very cool. That is really exciting, Moonrise Rabbit. Holy moly. Good for you. I'm excited for you. That's awesome.
And we had a question from Sparky. How can you tell the difference? How can you tell dinosaurs are related with no DNA? Is it similarities in bow shape and the age of the fossils?
Basically, yeah. Yeah, it's trickier. Like, it's a little bit more wishy-washy than if we just had DNA to look at. DNA is like a nice shortcut for that kind of thing. It's less subjective. But we have to try and infer what the genomics would be basically by looking at the morphology. You look at the shapes of the bones, you look at that kind of thing, and then you couple that with where they're from in time. It's also extremely important. So it'll make sense for them to be, near the base of the family tree or outside it, or that kind of thing. I hope that makes sense. Yeah.
It's difficult, Spark, and I think at least what the other paper was highlighting too is if you did have DNA, like even a little kind of DNA from these dinosaurs, you'd be missing a lot of segments. And so the only way, but let's say we could, just as a thought exercise, get some of those segments sequenced. What would you compare them to? So like that, Sparky, nowadays, it's how we do unknown samples. Dr. Soils, speaking of which, eDNA, Dr. Soils, thank you for the follow. Welcome in. That's how we do when, if there's a new species, Sparky, or even something that we think maybe has gone extinct as 100 years old, where there is some DNA degradation, you compare it against known sequences, where it's okay, we have a very well curated genome, meaning that it's not just a sequence. You don't just have a 400-page document with A, T, C's, and G's. You have here's a start site for a protein product. Here's another one. Here's a non-coding region. To that level, you have it. And then you compare your new animal to that close relative. So if there's a gap, you're okay, there's a gap here, but it's around this sequence that you have some sample DNA pulled in, right? And so you're putting it it's called blasting against something. You're doing a comparison. But if you were to take DNA from these specimens that we have, and again, let's just pretend for the sake of argument we can extract them, you have nothing to compare against. The birds, yes, ideally you'd compare, but it's too genetically distant.
It's going to be different anyway, yeah. It's not, that comparison's not going to yield anything useful.
And I guess...
You would have to have, DNA from 100 different non-avian dinosaurs from the Mesozoic.
Yeah.
And compare those in order to actually do anything, useful, I guess, yeah.
And maybe the only one that maybe we might be able to, right, is this Troodontid to your modern day bird, but even that, y'all, right, is a stretch, because you'd have to be getting a close enough relative to set living bird to the Troodontid that hasn't changed so much, where there's still some conservation between the two. But I think going back further, I don't think it's gonna be close enough. And our first time chatter, Dr. Soils, I like my geology in hundreds to thousands of years, still learning the millions. I like that, Dr. Soil. Welcome in. Are you a geologist, by the way? Welcome on into the community. So yeah, I think I hope that's hopefully answering some of your questions too, Daniel, the difficulties in some of these techniques. Even if we could get the sequence, there's just so many differences that you don't have anything to compare against. So y'all, it's not easy to do the stain to begin with, not because it's not easy to do the stain, but because of the sample type. And then we can't extract DNA, and then even if we could, the sequencing is difficult because of the size of the fragments you'll get. And even if you could get past that and you got DNA reads, you have nothing currently to compare against, especially because your DNA is fragmented. Dr. Soils, is a studier of the soils. Well, welcome on in. Great to have you here. But I still like this review paper either way, because it kind of highlights the opportunities that we still have and all the things to look for. That
sure. Yeah.
There seemed to be something unique about these kinds of bone that allowed there to be just conservation of the DNA. At least in the nucleotides. This isn't always happening in every single cell type, which is, I think,
definitely not. Yeah. Yeah, and I don't remember if it was mentioned in one of the personal comments things in the paper, but there's I feel a general, I don't know. The word on the street is that with some of these dinosaur specimens, where we can get what look to be soft tissues, collagen, cellular structure, even maybe DNA. The thing that might be kind of common across those is really, really iron-rich strata. It might be something to do with the iron in those strata, are the iron in the matrix, the iron in the surrounding rock, is maybe what's helping to preserve that organic material over millions of years. Whereas in rock that doesn't have that iron-rich quality, you know, the stuff just completely degrades and you wouldn't find any of it.
So it's again getting lucky.
Yeah, basically, yeah.
Yeah. Which is, I wish they would have gone a little bit more into that in the paper of like comparing the actual samples, like in terms of the discovery of the sample and what was the means by which they were discovered and the location, and like you said, like the composition, like kind of almost a field guide to a paleontologist who, like, if you're going in the field, Danny, and you wanted to have something to be brought back, if you kind of had a guide of like, okay, this is a good area where we might be able to get something out, I'm gonna be more aware and like prepped up versus just like you said, being gloved up the entire time.
I don't know if anybody's ever actually written that up and gotten it published, but so I used to work with Museum of the Rockies, and that's where Mary Schweitzer used to work too, and she's kind of a pioneer in a lot of this research. And what I remember is this idea that the bone has to be basically fresh. It can't be there on the surface. You've got to dig in and find it underneath, I don't know how much matrix, but a good enough amount, and then you got to get that into like a holding box or something like that where it's fairly isolated as soon as you can, in order to be able to hopefully get some organic material out of it. It's weird, in the Hell Creek Formation where I used to work, where like T-Rex and Triceratops and Pachycephalosaurus are from, sometimes you'll be working in a quarry and then you pull up some mudstone or something, and then you've got just beautiful bone there, and it smells organic. I don't know a better way to put it. It smells not like your typical dry mudstone would smell, but it smells like like an organism. And maybe it's something else going on, or maybe there's some sort of fungus or something like that we were picking up, but it could very well be that like that's just really iron rich sediment, and maybe there is soft tissue in some of these things, but nothing that I've ever collected has ever really been sampled for organic material like that. That's usually not what we're after.
Is there a way, if you're out in the field, to know that you're about to hit something that could have organic matter, and then what would you stop and like change equipment? Like what would one even do?
That's a good question. So I guess the only indication that you might run into something like that is if you're digging at a site where you've got bones on the surface, then you dig in further, and hopefully there will be bones that are beneath the surface that are kind of not exposed yet. Again, I've never gone out specifically trying to collect fossils for molecular studies, but that's what I would do. Go into a quarry where you're just kind of digging into the hillside, and there will be a bunch of fresh bone down there that's not been exposed to the air yet. It's still been in the same strata that it's been in for 66 million years or more, and then have stuff on hand, maybe gloves and like some sort of a container that you could just put things into really quickly, and maybe keep it refrigerated. I don't know, just kind of guessing at this point. But I do know at Museum of the Rockies, we used to have this big tractor trailer parked outside, or just trailer, I guess. I'll see if I can find you a picture. Mobile laboratory. But it was parked outside for years. Here we go. Image in new tab, and there is. Oh, why is that so small? Here we go. I'll put it in the chat for you. But Jack Warner, whom I used to work for, he got some funding to have this put together, so it could be towed to a site, basically, so that stuff could be worked on as soon as possible after immediately getting out of the ground. They could work on it under like controlled laboratory conditions, which is not the sort of thing we could normally do in the field. But I don't know if they were ever able to use this very much because it was just parked by the museum for years and years.
There was a video, right, that you've played before on your stream where Jack Warner is talking about one of his students was looking at like bone marrow. And I imagine that may have been what it was used for too, right? Like the immediacy.
Oh no, absolutely.
Yeah.
This is for Mary Schweitzer's work in the field. That's why there's a little logo on the side. It's Montana State University and North Carolina State University.
Oh, okay.
So Mary Schweitzer went on to teach at North Carolina State.
Oh, cool. That's
and
nice.
They may or may not have gotten NSF funding for this. I forget, but yeah. So it was definitely for that work, so that Mary could work on these things. But I don't know if it ever, maybe for logistical reasons, it didn't really work out in the end. It seems.
I just from what you've described to us about how to take out samples, like with the plaster jackets and the like, how on earth are you not gonna like? How are you gonna immediately be able to take something out? You know what I mean? Like it just doesn't.
That's the thing is that usually, you would have to have some kind of a bone that you're only going to use it for molecular study. You know, because you might well destroy it. You might destroy the morphology of the bone getting it out of the ground, because normally we've got to extract these things very carefully with, paper towel and burlap and plaster, and it can take days to do that. We're very, very slow and painstaking. But if you just have to tear that thing out of the ground as soon as possible so you can get it into the laboratory and start looking for biomolecules in it, you're going to essentially ruin the bone for any other purposes.
Yeah.
But that might well be worth it if you get biomolecules out of it. That's amazing. So it would be a very, very different way of collecting than we normally do. That would not be standard procedure, as we paleontologists are normally digging these things out of the ground.
And I think another difficulty is like if you're rushing to take one of these out of the ground, where like the crack site. You might lose a bunch of information, or you might not get the right one, right? Because or even sloppy salamanders, so you have the left and the right side, but what if it's the right side bone that has it, and now you've like busted it up, so it's like it's such a gamble, and I get you have to do it, like how else are you going to be able to pull it off? But it's just such a different method from like what you've told us about the very methodical, slow preservation above all else to just like and then just like throwing it into a container and going
yoink. Yeah.
But I mean it's exciting either way, and I liked it. That's why I love these crossovers too, is like hearing these differences about just even the logistical work, about how this is not straightforward. And I think hopefully, chat, we've illustrated that it's not straightforward on either side. Like the looking for the DNA isn't straightforward. Exactly, Moonrise, you have to yeet it out. Like, looking for the DNA isn't straightforward because of all these factors, but then even that initial sample collection is not straightforward. And so the fact that we have papers that do this, I think is huge.
I agree.
Yeah.
It's really neat. Like, I'm really glad that people like Alida Bayul and colleagues are working on this sort of thing and driving this field forward, because this is not... I don't have the temperament for this kind of thing. I don't have the background or the skills, or it's just a completely different way than I would be working, which is cool. But that being said, the lead author on this paper, Alida Bayul, I worked with her in the field in 2011. She was on the crew digging up the Yoshi's trike Triceratops specimen, MOR 3027. So I spent like the whole summer with her digging up that Triceratops, and now she's on the cutting edge of molecular paleontology, which is pretty darn cool.
That's really, really cool. Was the entire summer that one specimen?
Pretty much, yeah. That was, I think, late June through mid-August that year.
Wow.
We were digging up that one specimen, and that wasn't even its skull. They took the skull out the previous summer. And
most of the skull anyway. And so we were just digging up the postcrania. This is an animal like the size of an elephant. It's a really big Triceratops.
Wow.
So yeah.
Is it again in terms of time? Just because you were saying you had to do things to make sure they were kept and preserved and like not damaged, and that's why it takes so long.
Basically, yeah, and we just had a lot of the skeleton there. It was fairly complete as far as Triceratops go. Usually with Triceratops, you just get like part of a skull, and usually that's it. Occasionally you find other bits and pieces, but having an associated skeleton, yeah, it's really important to be able to get... Well, it was really important to get the whole thing out in this case because it might be a new species. Hasn't been named yet, but it seems to be intermediate between Triceratops horridus at the bottom of the Hell Creek and Triceratops prorsus at the top of the Hell Creek. So it could be a good example of anagenesis, like one lineage just evolving and changing over time, because it's got a bunch of characteristics in common with the Triceratops from the top and the Triceratops from the bottom. And of course, we found it right in the middle, so it fits just about perfectly in terms of that story of evolution of Triceratops during the Hell Creek.
And it also brings up then the difficulty you said, like, when do you know to cut off a species that's just like undergoing natural selection but now has become a new species? Like, where is that threshold? It's interesting that it's not already named. You would think after over a decade. Naively.
It's not really been published on at all, though. I mean, it's been featured in a couple of different publications. It's Nosehorn in particular, I think. But there's no monograph on this critter yet. There's no detailed description or anything like that. And at this rate, I kind of wonder if there ever will be. At least while this current team is there in possession of the specimen. I don't know. I really, really want to see this thing published, but hopefully someday it will be.
Yeah, that seems really surprising, like unexpected that you'd have something so massive, and then for so long undergone no kind of real big publication. But like you said, there's a lot of difficulties with publication anyway.
For sure, and I think it might be. You and I probably both know older professors who would, they would get tenure and then just hardly ever publish again. That's a thing that happens sometimes, and there might be a number of reasons for that. I'm not trying to condemn anybody for that, but it happens sometimes, and I'll just leave it at that.
Yeah. But it's really cool that she has this connection to the field work, what you were doing, and then now is looking at this molecular side of things.
Yeah, this is when Alita was a PhD student under Jack Horner, working on molecular stuff, working, I think, alongside Mary Schweitzer and other people at the museum. She has also done a lot of work on dinosaur growth, like looking at dinosaur histology, growth patterns, that kind of thing. And it seems like recently she's doing that as well, but she's also doing molecular paleontology. Alita's doing some cool stuff.
I can see that kind of going together. Like the histological
a lot of microscope
side.
work.
Yeah. It's like, and especially like how many experts are you going to have in the world?
Exactly.
Like you've said, it's such a small field to begin with that it's not going to be something straightforward and easy to really pull people in necessarily. And so, if she is the premier place to go to learn this.
Exactly.
Yeah. So yeah, just a little wrap up on the paper. I like too that they put a little bit at the end of like, here's some examples of what might contamination look like. And then having that comparison to like, here's the bacterial film and the collagen and the other stains, and like, this is why when we looked at these like alleged dinosaur samples, we didn't see these like remnants of what contamination would have been like, and therefore we are pretty confident that this isn't just a guesswork, that it's from the sample of the dinosaur, but like compare of what pollutants should already look like.
Exactly.
And it doesn't look like that.
anytime you're reporting on biomolecules from a dinosaur that's tens of millions of years old, the first question in everybody's mind should be, could it just be contamination? And so you've got to mount a pretty robust defense against that and say, like, no, this is not contamination because of X, Y, and Z. And I think they did a pretty good job of this. And it's funny, like, when some of these first findings were announced, back in, like, the mid-2000s, around, like, 2006, Mary Schweitzer was reporting on finding soft tissue in a Tyrannosaurus. You know, like little bits of collagen and blood vessels and stuff like that. Basically, nobody wanted to believe it, including her at the time. She's like, I don't believe it. Like, let's do it again and see if we get the same results. And to me, it's like a beautiful encapsulation of how science works, where we might be really enthusiastic about some result that we get, but the cooler a result is, and the more extraordinary it is, the harder you got to work to prove that it's actually real. You can't fool yourself with that kind of thing. There's that saying that, like, the easiest person to fool is always yourself. So you gotta really fight that urge and be like, you know what? I don't believe this myself. Let's do it again. Let's try and figure this out. How could I have made a mistake? You know?
I think what is the quote? It's extraordinary claims require extraordinary evidence.
Yeah, Carl Sagan. I think that's his phrase. Yeah.
Yeah.
And it's a good one. It's kind of at the heart of science.
And it's hard too, because of, like, these kinds of samples aren't easy to work with to begin with, and so knowing what contamination is here, when you don't kind of have a book to go by of how to ID contamination, is really difficult. Because like there was, I think Lita has mentioned this before, but there was this really, really cool paper a couple years ago when we were in grad school, and it was where tardigrades got their protective genetics from. And the paper hypothesized it was horizontal gene transfer. So jumping from one organism into the tardigrade, and then the tardigrade just acquired all these genes to give it, like, dehydration protection and heat and pH and, like, you know, everything. And it was, you looked at the paper like, man, this looks, you see the DNA bands, you see the sequencing, this looks great. And Lita presented it, like, at Journal Club, and everyone was all about this paper, and, like, a year went by, and they're like, yeah, that was all contamination.
Nightmare! Yeah.
So, Lita, I know there were people coming up years later to her like, I remember this paper that you presented at Journal Club. That changed my outlook on tardigrades. And Lita had to be like, yeah, it was all contamination, I guess.
That's why we test things in science, you know? This is why. It's, yeah, and that must have been heartbreaking for the researchers to realize that, oh, shoot, they goofed up and contaminated it, or something happened to contaminate it, and, like, their beautiful results nullified.
Yeah, and where it's like it logically made sense, like, and there was a lot of good data and replication there and everything. It was just the same methodology had gone wrong, and that led to the contaminant throughout every sample. And so it was like that one starting point, you know, all of it. So I say, you know, hats off to them here, because it's much harder, I feel like, to, you know, there's no handbook on what you're looking for. Each one of these test cases is different. Like, I mean, Danny, even, like, each one of these stains, right, is different. There's caveats to these stains in modern living cells. How are you going to apply that to a dinosaur? Like, or, you know, like, this is from this hip, you know, it's just, I don't even know what to do. And so it's really remarkable that something like that is able to be identified and found. And so, like, I don't know, I was really excited by this study, and I hope it leads, like, it's unfortunately an older review, and so it's not from, like, a big news story or anything like that, but I hope it is pushed into more people finding it and, you know, being excited by it as well.
Yeah, I hope so too. This is such a neat field, and I'd love to see more progress in this. As little as I understand it, I'm so glad I was able to talk about this with you, because, you know, this is kind of your area of expertise to a certain extent, much more than it is mine.
That's why I
deal
love
with fossil organisms here, but, like, I don't know anything about these methodologies. This is not.
Well, it's kind of like a black box, right? Like, same when you tell me, like, yeah, we collected dinos' bones for the field. Like, I don't know what that means. And then you go into the details, like, oh god, that's like a really intense. You're like, no, it's fine. We do this, and it's like, it's a cool back and forth of like, of the differences in our fields, what we think is difficult, and kind of conveying that to the public too. And that's like the beauty of these crossovers.
Yeah. So cool paper. Thank you for picking this. This has been a lot of fun, and I also hope that this spurs further progress in this field, because I'd love to be able to say, yes, we do have DNA from dinosaurs from the Mesozoic, and this is what it looks like. But yeah, a lot of challenges with that, and
before we move to maybe the proteomics paper, because I feel like it's in vain here, we have a question from Squall. I'm curious about papers. Aside from groundbreaking ones, what motivates a scientist to publish one? I know there's prestige associated with papers, but a not interesting or forced paper could end up being negative for the scientist, I imagine.
If you can get it published. If it's not interesting, good luck. Yeah. You publish a lot more than I have, Dylan. I feel like you should.
So, I think Danny and I publish because we're one to advance scientific knowledge, right? That's like the purest reason to publish, is you want to have made an impact in the world's knowledge. And by discovering something, whether it be groundbreaking or just a next small step, you've added to the collective intelligence of humanity, right? I think that's like the joys that we have in publication, of spreading that knowledge. But honestly, Squall, it comes down to money. If you don't have publications, you don't get funding, and then you can't have any more publications, and then your scientific career is dead. Same thing with hiring. You won't get hired as a postdoc, a graduate student, sometimes a professor without publication. So, like, that's your currency, is publication. So when you say not interesting or forced, as Danny said, that's difficult because there's a lot of papers that won't ever get published with negative data, which is, it's hard because, like, what if I do an experiment that doesn't work? What if there are 3 other scientists that are planning to do that the next year, and they're wasting grant resources trying it the same way I did? They'll never know.
Realize that it doesn't work. Yeah.
Exactly.
You can't warn them about that through a publication and say, hey, we got a negative result. Here's the paper. Because no journals want to publish that, because a negative result just doesn't sound like it's interesting, even though it might actually be.
Yeah, or at least, and it is contributing to general knowledge, right? Like, you know how something didn't work, that's huge. And it is not a fair, it's not a money grabber for the journals. Yeah. Nautical Pumpkins, thank you for the follow. And Gemseer, welcome in. So there's, I mean, that's the big part. And a forced paper, so forced papers in my mind are, if you have a finding, like, let's just say, like, I examined this behavior, that and actually can be a paper. You can publish that. I swing on the opposite effect of, like, I want to figure out the genetics and the neuroscience behind this behavior and tie it all back. And that actually, I have papers that could have been 6 papers, but instead of making it 6 individual ones, I try to string it into one collective story, and that gets you a higher tier publication, which also can correspond to better funding or job opportunities. And so, yeah, it's this weird currency that we have as scientists to get jobs and funding, and not, it's kind of gone away from the beauty of advancing knowledge in the world. Would you say that's at all a fair assessment, Danny?
Yeah, I'm trying to think of some sort of an analogy that might make sense. And the thing that popped in my mind first was, like, being a film director, where it's like, in the same way that scientific publications are the currency within science, they're like, how you judge a scientist's work is by her publications. And that's what gets you jobs and funding and raises your star and everything else. Maybe it's kind of like, like movie directors making films. And maybe you can make a series of short films, but instead now that doesn't really. I don't know how well this analogy works.
You got a few Oscars, and so now you're like the big ticket item, and they'll give you a ton of money for that same role that you could have otherwise only gotten $5,000 for, you get 5 million for.
Maybe,
Maybe,
yeah.
and
Yeah,
then-
if you were able to get a Nature or a Science paper or Cell or something, yeah. Yeah, it is like having a big blockbuster film. Or, I don't know. The analogy doesn't work super well, but...
The computer
Yeah.
science Sir Caffeine, it depends. There's a lot of what we call bioinformaticians or bioinformatics as a field where there was one of my mentors, he was exclusively bioinformatics, and he gave out every year a lamprey award, because the lamprey is like,
Lamprey Award?
yeah, so it like leeches on, right, to another animal,
Yeah.
and like,
It's a jawless fish. They're really cool, like Nathan fishes, yeah.
yeah, and so he would give someone who the lamprey award, who was the most like leached off someone else's work, like the big fishes, and just ate it away. Because what you can do if you're out of funding as a bioinformatician, if you have a computer and a data set you can publicly download, you can analyze stuff. Like, that's the beauty of that skill set. And there are journals that'll take that, like not even predatory ones. Like, there's always the predatory ones, but there are ones where it's like, hey, I analyzed this data set and I found it looks like there's an association between this phenotype with this gene. That's a paper. And so for bioinformatics, if you want to get a PhD, I think the average time for that doctorate is 3.5 years, versus a molecular like PhD is like 6 or 7 years. Mathematician Sir Caffeine, I have no idea. I'm like, I don't know how people get published on that as well. Like, do you just sit and solve an equation on a whiteboard? I'm sure there's more nuance to that, but like, yeah, there absolutely must be something difficult there, and it's a really cool field, but not at all easy. Yeah. So Danny, do we have time to start on the? I think a similar.
Let's do it. Yeah, I've got about an hour here.
Okay, cool. Do you mind if we start on the eggshell one, because I think it's similar in this the sense of like, right, breaking down preserved specimen and what it means to be like these methodologies that aren't able to work, right? Like I think we're on similar lines with the DNA versus the protein side of things.
Yeah, I think so.
Okay, cool.
Yeah, let me get this pulled up. Yeah.
Yeah, this is a pretty exciting one too, I felt.
Yeah, I wasn't actually able to get hold of the full paper. It was just a preprint, I think, that I got, or like an unformatted version.
Do you want me to send it to you, Danny?
Actually, if you wouldn't mind, that'd be great.
Yeah, of course.
Yeah.
Do you want it right now?
Yeah, don't worry about it. It's gonna take a minute. I could just look at the preprint.
It'll take what, 2 minutes? I have your email like on easy access. We're all good.
Oh, okay. Well, cool. While you're doing that, let me just say that. Oh wait, no, shoot, this is a different paper. Never mind. There's the other one that 2 of my roommates from Montana are actually authors on the Bergman's Rule paper.
That one was really cool too. And don't get me wrong, I didn't not pick this because I didn't think that it was exciting.
Oh no, of course. No, but this does have more relevance to what we were talking about.
But I do
actually looking for biomolecules in dinosaur fossils that are tens of millions of years old.
Yeah, and the Bergman rule too sounds really interesting, just as an aside of like how it corresponds to modern day animals too, and like the impacts of it. Like I like to take away a lot of like, what can we do that'll be meaningful, like in fossil science, that influences knowledge of animals today.
What perspective can we gain from the fossil record that we can't glean from just looking at modern critters?
Exactly.
Yeah.
Let me just one final grab, y'all. I'm just downloading this PDF.
Nice, thank you, Belinda.
Of course, yeah.
Sir Caffeine wants to know why does it take so long to get a molecular biology doctorate?
That is a great question.
Big part of it's going to be the experiments, right?
Yeah, the experiments and stuff goes wrong. So when you're doing a molecular experiment, like for example, when I was doing the RNA extraction study for leafcutter ants, no one had ever done that before. There was no reason to suspect that it was going to be difficult, because why would it be? It's like you do DNA, RNA dissection all the time. And then there was just this unique feature of the animal that had this overactive RNase, which breaks down RNA. And to figure out how to get around that took 6 months. And so you can imagine an experimental setup where it's like, hey, this should be straightforward on paper, but then you fall into a situation where it's not as clear-cut as we originally thought it to be. Does that make sense, Sir Caffeine? So you just come across variables that, for better or worse, they're not easy to circumvent, and the papers just are not easy, like, to replicate even. Like, there's been studies where it's like you take a look and you're like, oh, this is an easy step-by-step protocol, and then you get to the protocol like, wait a minute, there's like 30 steps missing. And it's just because there's like implied pieces. One sec.
Yeah, similar things can happen with paleontology as well. Like, I know my old crew chief, his doctorate took 10 years to do. And that was because he got a little too ambitious with it, maybe, and he was looking at, like, the evolution of entire dinosaur groups across the late Cretaceous of North America, and that was just one of the chapters in his thesis. He had, like, 5 other ones. Yeah, sometimes if you're looking at something really big and grandiose, with a lot of different ideas, it can take a long time to gather enough data and to analyze it. Yeah. So not all PhD theses are equal. Some of them require a lot more work than others.
It should be in your inbox, Danny.
I'm seeing it here. Yeah, perfect.
And Sir Caffeine too. If you're working with living animals, there is a phenomena where they don't behave. Like,
yes,
you have an expected behavior for wild type control, and they don't do what you expect them to do, and it turns out that it's because, like, this is a real life example, Sir Caffeine. We had a collaborator, lose a year because the janitors changed the cleaning product, and the fruit flies could smell the change of the cleaning product, and that affected their ability to form the kind of memories that they were studying. And it was to the point where the students... Bacchio, thank you for the tier 1 sub. Welcome the heck in.
Bacchiotomy just subscribed for 3 months.
And a piece of raw science and a partner plus point. Thank you, Bacchio. So yeah, like, we had... and Squall, thank you for the follow. That student who was leading that experiment ended up not, like, her clothes... She bought all new clothes. She bought, like, changed out perfume. Like, like, body wash, hair wash, everything under the... Because everyone's like, there's no... Like, it's something we did. And it was as simple as the cleaning product was changed for the floor mopping. And it's like, how do you account for that? Like, if you're trying to replicate an experiment in a lab across the country, still even in the U.S., like, not even a different country, and that isn't usually in the protocols of what you use to clean the floors with. Like, I have never seen that in a protocol.
Right.
And that, like, that's where, again, you go from, like, you know, a 3-month experiment to, you know, very easily a couple of years. Yeah. Is that, like... Yeah. And Real Man says, is that, like, a situation where the animals repeatedly go into traps because they'll get food, a safe place to spend the night? That's, yeah, like the Pavlovian training. So you have this associative formation, associated memory. That's why, like, when working with mice, the experimenter isn't the same person as who does the feeding, because the mice will even learn visually to associate you with a positive reward. And then if you're trying to do an experiment that's about negative memories formation, you better not be doing that experiment, because then you're gonna overpower. Like, there's already some kind of memory that's gonna be possibly overpowering the new memory you're trying to instill. So there's a lot of difficulty there. And telling Timmers how cognitively smart any animal is super difficult, because they are smart for what they evolved for. Right, like
exactly.
a fruit fly.
Point. That's a really good point, Bullet. I love that.
Well, it's hard, right? Because you're like, surely a bug is stupid. It's not as smart as I am. But, like, what is smart and what is intelligence? Like, a fruit fly can remember a parasitic wasp that it saw a week ago and has changed its behavior accordingly. I mean, on if that was on our time scale, that would be you remember like a threat from a decade ago and are still remembering to alter your behavior to account for that potential threat, like in terms of like
Sure.
lifespan and timelines. And that's
Yeah.
a pretty powerful memory, but yeah, I mean, it's not the same kind of learning and memory that we have because that's not what they evolved for.
Yep. It's like a woodpecker remembering where it placed literally thousands of different acorns, and then also being able to migrate across, halfway around the world, and like not get lost during its migration. And then, I know people who get lost even with Google Maps on their phone, and there's different kinds of intelligence, I guess, and we tend to have a very like human-centric view of intelligence. But that's the sort of, I don't know, what you consider smart might get you killed as a woodpecker or something. It just doesn't work under those circumstances. So there's so many different kinds of critters that have different kinds of intelligence. They've evolved for, different niches. And so trying to like compare apples to apples when we're talking about something as vague as intelligence is really, really difficult. It's difficult to define your terms, first of all, let alone like actually run experiments and stuff.
I think that's an additional layer of complexity, that the term cognition and intelligence might be different in humans that we know, but applying that to an insect or a mammal or a bird, those definitions are inherently going to have to change. Like, what is cognition in that animal? Like, supposedly an ant is self-aware. That would mean that it should have the ability for cognition. I'm not convinced by the experiment, but if you do buy this like mirror experiment, then it is self-aware and therefore it has cognition.
Oh wait, they've done like the dot test on ants and they passed it?
Yeah.
Really?
Well,
I didn't know that. Holy cow.
That's what the paper claims. I have like a large eyebrow raise at it because it's not. It doesn't. The geneticist in me is like, oh, I don't know if that's a real thing. Like, even like from an ecologist perspective, it's a little dubious in my mind. But like, there was like they put a dot on the mirror, and when the ant looked at itself, it tried to swipe the dot that was on itself.
So it's the idea is that it's recognizing itself in a mirror. Like that's what the authors are claiming, I guess. That's wild. I've never heard that for any kind of insect before.
Well, there's a second paper in fruit flies that claims the same thing, but they did mathematical modeling based on behavior to suggest that not only are they self-aware, but they can visually tell individuals apart. Including if they look in a mirror as well, that is not different. And so that was a much deeper in terms of the mathematical modeling. I still raise an eyebrow at it because I'm like, your model is only as good as the data that you put in, right? So if it's garbage in, garbage out. I'm not saying the data in was garbage, but it's just so hard to figure something like that out.
Yeah.
But you can't ask the animal, right? So it's like you're control and awareness in one animal might not be the same as awareness in humans. So it's like these definitions I feel are just really, really difficult. Yeah, exactly, Dame Karen. We're just skeptical on all these fronts. Like, we don't. Lita likes the redemption on only when she's here, but she made this bunny rabbit that comes across the screen. It's a skeptical bunny. It's not a convinced bunny rabbit, so there it goes. And it's like the same principle, right? It's just that it's a great idea, but something that doesn't quite necessarily add up there.
Yeah.
And so yeah, sorry, Danny. Well, now that we have both had the paper, do you want to prime us into this?
I didn't even realize that this many authors. This is really funny seeing it formatted like that. Let's see if I recognize any of these people. Paul Sereno, Xi Chen. Yeah. So, one of the things that I thought was really interesting about this is that they're not taking samples of eggshell that were specifically collected to look for any biomolecules or anything like that. These are just bits of eggshell that somebody on the team had from when they were a kid that was like purchased from a rock shop.
And so that was kind of wild to me. But basically, yeah, they're testing these for biomolecules. Goodness, I read like 6 papers today trying to get ready for this, and it ended up being more confusing than anything. It was calcite was kind of like the silver bullet here. So they're saying that calcite, this material in the eggshell, might actually have like a certain degree of preservational advantage for preserving biomolecules. Was that what you got out of this, Tibble?
Yeah, it was like, and they were surprised too. Like, it did seem like a very unique finding to be able to pull something like this out. And it's, and yeah, Gojira, the contamination actually they do get at, right? Because that's an immediate question that you might be able to bring in. It's like, okay, there's this rock shop, and there is a kid, and their home, right? There's just like, probably looking at, like, who knows, right? But it was, they found this racemite amino acid. So amino acids have 2 different forms. There's actually on this this kind of image, you have the left and right-handed versions of an amino acid. And as you have decay happen over time, you go from one form to another. And eventually it evens out 50-50 is the idea of it. You go from this left-handed form to the right-handed form, and then there's a constant shift, but it's evening out at 50-50, and that's a way to tell. It's not a DNA, but I guess a protein was the term, right? The ancient levels of protein, when something's kind of hit at its like way past half-life, and it's a way to date it. And so Gojira, if there were contaminants in these samples, and correct me if I'm wrong, Danny, in my understanding, you should have seen not a 50-50 ratio of these amino acids of like this racemite form, but you would have seen a peak in the left-handed form.
That's what I got too. Yeah. Yeah.
And so I thought that was a very clever way of the workaround of like any potential contamination that could have happened.
As a fellow laboratory scientist to the authors here, what did you think of their? I don't know. When I was going through the paper, the methodology, most of that just sailed directly over my head. There were very few like points of recognition or like, oh, I grasped this part. It just kind of washed over me, and I hardly remember anything from it. As somebody who works in a laboratory, you're much better suited to talk about this than I am. What did you think of their methodology?
So, my first thing was, why is this not a Nature paper?
Oh, really?
Like, before I read the methods. as I went.
Okay.
And that's not a knock on them, it's actually going to be a knock on the results, like, of what ended up happening. Because I feel like if you're able to extract via a mass spectrometer protein from an ancient source like this, like, that's huge and groundbreaking, right? Like, that is a big deal. Buried in the discussion, I think it was, they had this little caveat that they were able only to find 4 amino acids out of the over 20 amino acids that exist. And that was, only 4 amino acids. GLX, GLY, ALA, and VAL, so I think that's like glycine, galantamine, alanine, valine, show clear, consistent evidence of survival in the various treated samples. And so the reason that is an important caveat is normally, like, these figures, Danny, are very, very powerful right here. So, like, let me break down this figure. So this is what we call an LCMS, so liquid chromatography mass spectrometry. And what you do is you liquefy a sample. So I did this with histones. Remember, that's what we talked about, Chad, actually in a previous crossover with the DNA wrapped around those little beads on a string. You can isolate those beads and figure out how they're modified. And so you separate them out from the DNA. So here they separated other biomolecules out from the protein, so you're getting the pure proteins. You liquefy it, you put it into a machine in a very concentrated form that shoots an electric current through it. And when it shoots an electric current through it, it aerosolizes it, goes into this mass spectrometer, and then you analyze basically the molecular weight of things as they're being detected by the machine. And so the length of time that a sample is in the machine will give you a certain resolution of the material in your sample. So if, and here they went with this, they did a longer time, so actually an hour is considered like a long time in this machine, which I did not know originally. So if you already know what you're looking for, you can tell it to filter by the atomic weight, and you can put in the sample for 5-10 minutes. The height of the peak tells you how much there is in the sample of this material. And so if you know what region you're looking for, you just tell it, okay, 5-10 minutes. You look at the peak intensity, you're like, okay, it's really, really high or really low, and you're done. Here, they're looking for everything possibly that they can isolate. Because, right, we don't know what we're going to find. And so you put it in the machine for longer, so it's a custom protocol for an hour. And then you're getting all these different peaks of materials that you didn't know what you were going to find in there. So there has to be a system for them to deconvolute this data, because you otherwise just get a sheet of paper out with these peaks and you don't know what it means. And so the hard part is when we do RNA sequencing, it's standardized. DNA sequencing is standardized. These mass spectrometers, every time you do a mass spec experiment, you're writing a custom protocol to detangle what exactly each of these peaks are. So when we did ants, that was different from the histone. That was different from my friend who was looking at beaver proteins and comparing them to naked mole rat proteins. Like it's like there's all these like very special things you have to do, because while the machines for mass spectrometers are very good, there's no software that standardizes them. And so I think inherently the methods are difficult in these studies, because you're basically, they're building a database from your chickens of what you know to extract. Like, this is like a modern day sample, this is what we find, this is what we predict to find, right? So they have a database built from that. That's how they identify what the peaks are. They then, if I'm again understanding correctly, heat treat chicken eggs or other modern day bird eggs to simulate damage.
Sure, diagenesis, yeah.
Put it through the machine again and see how those peaks have changed, to almost get like, here's what we will predict that the dinosaur will look like
after fossilization and everything else, yeah.
And then they put in the dinosaur. And so what it's cool is, like, in some of these peaks, we can see some molecular matches, like the 2,4-dimethylphenol, is the same peak between the two animals. But you can see the intensity of the peaks is different. So where there is this like indole, or the 3-methyl indole, that peak isn't even noted right here in this one, and that's partially because you're only pulling out certain amino acids, so you're going to be missing a whole cluster, and then the other thing is you are getting degradation. So it's the amount that you're pulling out is going to be different between organisms. With the state of, fossilization, you're losing a bunch of amino acids, and then just also what you're able to identify. Like, you're still using a reference, like with the bird, to be able to compare back to. So I agree with you that I thought the methods were a little bit convoluted, but I understand the logic behind why they did it the way they did. Especially, like, with our crossovers of, like, bird, let's take the bird and then, like, take the egg and, like, simulate damage to it the same kind as was with the fossil. And then we do the fossil and see, okay, stepwise, how do the peaks compare? So I think that logically makes sense. It's just then you've lost like half the data just as a function of that preservation and how it works. I think that's why it's not as high of a tier, because you're only pulling out 4 amino acids, and not
Gotcha.
getting everything out of it.
Would you expect to have 20, like every existing type of amino acid in a sample like this, or would there only be naturally occurring certain kinds in an eggshell?
So with the bird data, so with like the living bird eggshell that they took out, you have all the amino acids because they're going to be different part of the eggshell itself, but in different quantities. So you should still get low levels of everything, at least incorporated into the molecules that you put out. So like, you might get a really long string of carbon, and there is this amino acid hooked onto the end of it. Like that's enough to get you a peak, because what ends up happening is if you have a really long chain of carbons, that the time that you go along here indicates the size of the molecule.
Okay.
And so you can imagine where if you have a string of carbons and then you attach something big to the end of it, even if it's just one big amino acid, that's going to shift the peak down.
Gotcha.
And so you have like each of these little peaks, even though they're not all marked, have some of those different populations on them to differing amounts. And so maybe some of the low amount of ones you might not predict that you would get from a dinosaur. This is just my naive guess because of degradation over time. But
Sure.
It's surprising that it's only those 4.
Okay. Yeah.
And so I don't know if that I don't know if it's like that they didn't get preserved or don't preserve as well, or maybe it's something
Or
where
it could be a problem with the methods themselves.
well, maybe it's one of those things that the tech just hasn't caught up yet. Like it's brilliant
Sure,
that we
okay.
can do this kind of tech, and maybe in 10 years it'll be sensitive enough to pick up other populations and species like of the amino acids. And it's not, like there wasn't really clarity to me as to why it was only those 4. It was just that this is what we find.
Gotcha. Okay. Huh.
But it is, I think, really cool that there was the ability for us to see something like this. And then here to get at, I think it was Gojira's question about the ratio. So here they're looking at those ratios of the different forms of the amino acids. And as you're aging the samples, you're going to predict closer to 50-50. And so just an easy way to look at it, right, is if this is your ostrich, right, here you have a gap in the proteins where this population has a really, really high value of this image of the protein, and then the mirror form is much lower, versus in our dinosaur specimen, see how the lines are overlapping essentially. Like, I know they're not on top of each other, but that's within the realm of error. And so because of that overlap, right, that's at that 50-50 mark where you expect, like, that's your age sample.
Gotcha. Okay.
And so like all of their dinosaurs, like, I mean, this Chinese hadrosaur is remarkably on top of one another, right? Like it's beautifully matched, like showing that 50-50 equivalent. And actually you can see the few amino acids, right, that are present, right? There's the GLX, the GLY, the ALA, and the VLA, and everything else is just tanked in terms of detection. Like, it's just not there. And so I think that's a really interesting way of looking at it too, is like each of these dinosaur graphs only have those 4 amino acids, and then here with these modern day birds you still have other amino acids that are also like phenylalanine, alanine, I forget, ILE, I know doesn't actually start with an I of what the shorthand is for it, but all these other amino acids too, you still have populations of. Sure, they're lower, but they are detectable, and versus just like the straight up 0 across multiple different dinosaur eggs.
Right. Well, based on what you know about different amino acids, given that I know nothing at all about them, would you expect some of them to be more resilient than others over time, or through the fossilization process, stuff like that? Like,
I guess. Yeah. Oh, that's a great question. I'm just shooting from the hip now, to be, because right, it's not my area of expertise, so I can hypothesize, but, and maybe, I guess that's the beauty of these kind of crossovers, Chad, is there are going to be questions neither Danny nor I know, but our expertise puts us at, we can maybe predict. Tommy, sleep well. A good night, Professor Ploticus. So I would predict that the charge of the antibody or of the amino acid could be at play here. Of like if it's a really negatively charged amino acid, if it's really positively charged, because then it's more likely to interact with a metal in the soil. Like you're saying, like maybe the iron deposits would like neutralize some of those charges, and I don't actually know what direction that would go, if it's more likely to be better preserved or worse, if it's maybe like the charge is neutralized, but I can see it binding to something else. And then maybe if it's in a bound state to something else, it's not any detectable anymore. So that's what I can kind of see as some of the differences between it. Is like the charge is the big thing for me that is popping in my head. But that's not to say it's even accurate or it's the only thing.
I never even think of that. That's interesting. Yeah.
And JP, welcome in. Wombat, I think, also sneaking on in here. Hope y'all are doing well. Welcome to Hacking. I've been got. Thank you, Turgus. Danny, I'm gonna have to make it so your camera also drops down, so we both can be gotten at some point.
There you go. Yeah.
The basement pool is very nice, Sparky. I was on the phone with insurance part of the day today. So, the basement flooding. It's a whole thing. But what did you think of this paper? In terms of like, is this exciting to you? Like, what was your feeling on it?
I was really excited to read the abstract and read the press release. Well, maybe half of the abstract, and then I was just a little bewildered because I understand so little about what's going on in the methodology and everything else. So much of this just sailed over my head, which is kind of neat in a way, that you've got, something paleontological like this that is just completely outside of my understanding. To me, that's almost kind of exciting, because that means there are people who are working in the field will understand, like, it kind of shows a broadening of the field. Paleontology is already an extremely broad field, but moving into different avenues that I have zero understanding of, I think is pretty cool. Like, that there are researchers who are able to glean an understanding about stuff that I cannot even begin to grasp at this point. So it was humbling, I guess. And that's my main takeaway from it, really. I was eager to hear what you had to say about it, which is why I picked it.
It's a similar vein for me, too, where it's like I understand bits and pieces of it. Like, I've done mass spectrometry, so I can tell you, like, how to interpret the data. But then it's like, I'm stuck on the other side of it, where it's like, all right, but, like, sample extraction on a fossil... like that just seems really difficult. Like, what's the database that we're planning against? And, like they accounted for that in the chicken, but is that the appropriate one to do? And it's like these kinds of other questions of like kind of how you've said of like we're comparing to the modern day ancestor as best we can, but what's like is that the appropriate comparison? And I guess I would have liked to have seen more modern samples, because it's like. In this analysis here, like in this panel A, right, we see the 4 amino acids that are represented in the dinosaurs quite nicely. But we see other ones too. To get at your earlier question, well, what's the most representative? If we had more birds, I know it's just a snapshot. And like you said, it's a small population of the birds that are around today. But what is the representative bird egg in terms of protein composition?
Right, yeah. And I wonder how much that would vary, too. Like, I don't know. When I was at Montana State, some of my friends and colleagues worked on dinosaur egg microstructure and composition and that kind of thing. And the number one takeaway that I got from talking with them about this is that dinosaur eggs are weird and highly variable, and they might look very, very different from one another and have different microstructures and compositions, even in eggs from two different dinosaurs that are in the same family. They're very close relatives. They might have wildly different stuff going on with their eggs, and I would expect it to be the same way with modern birds.
So when you say different, what features were they looking at to get those differences?
They were looking at a lot of morphological stuff. So like the size of the pores, and when they would do like histology of the eggs, like the cross-sectional stuff, like different, I think even like different crystalline structure and stuff like that in the that lattice work that makes up the eggshell. It could be highly, highly variable, and I wouldn't be shocked if that can even vary, like in eggs from the same mother dinosaur, just like season to season. Maybe that would vary. I don't know.
That's what I was thinking right away too, is the temperatures, or even tying to the other paper that you picked today, like the latitudes, right, and like the different locations there, because you'd predict, right, if the pore size, like that must be thickness of the egg, and and this is actually one of the figures from the paper where they were looking at some of those cross sections and comparing between, you know, your modern day birds and your ancestral dinosaur, and like seeing like what was the thing that's preserved, how it looks like. Yeah, I maybe naively predict that there would be variance in birds today, but I don't know. Is it something where like the amino acid composition is near set because it works so well? You know, like evolution figured. Yeah, I don't know either. But like, did evolution figure out a way that this just works? Like maybe the oxygen exchange and like the support structure is like, yeah, there's differences in amounts, but essentially it's all the same amino acids found. And maybe like in the modern birds you see all of them, and then the dinosaur sample you only see those 4 because those are the only ones that are preserved, and not because modern day birds gained them later, right? That's the other thing that I want to know is like,
sure, yeah.
Is it that you can't detect those, or is it that this is an additive that your modern day birds have evolved to like express these at detectable levels?
Yeah. Huh. So you would have liked to have seen more different kinds of, you know, more modern bird taxa represented in the eggshells?
I think it would have been a nice thing to see, and then just like damage those as well, and just see like if you damage the eggshells, like in this one bird example, they have heat treated specimens marked here in the red, and you do see that like at least one of these specimens, like the glycerin, is gone. So like normally when they're not heat treated, you see them, and then the heat treated, like measuring and demonstrating the effect of fossilization, got rid of the detection of that amino acid. Like, and I know this is probably not the case, but I just would want to know, is this just a fluke of this bird in reacting to heat, or is it actually representative? Because like with the dinosaurs, I buy it. It's like they did multiple different dinosaurs, and so it'd just be like what you're comparing to. I think it would be nice to have additional ones, like maybe how Dame Karen said, an emu, for example. I don't know how easy it is, Dame Karen, to get a hold of some of these specimens, especially if the bird's endangered or political boundaries.
I've collected emu eggs before for experiments. There are emu farms around the U.S. where you can get them, yeah.
Okay, cool. So like an emu then, right, would be great to do. And, and then you'd maybe have specimens to heat treat and not heat treat. And I think like that extra representation would be really cool to be able to have. And then, I mean, Celtic, I think it would be neat to have the reptilian side of things, like with the crocodiles. I think, as kind of we talked about earlier tonight in terms of the phylogenetics, it probably wouldn't be the closest relative, but it could be...
Yeah, and also crocodiles have a different kind of eggshell too, if I remember correctly. It's more leathery than these like hard-shell dinosaur eggs. And so you're just going to have a very different kind of structure and makeup, I would imagine.
I guess the only thing that it would be, where it could be beneficial, is if you showed that there is like a loss of the same amino acid upon treatment. It's like almost showing conservation, that this breaks down as a function of fossilization, maybe. That could be conserved, right?
Yeah.
You.
are wholesome as fit.
Oh, thank you, Spectre Flood.
But yeah, I like the paper a lot, even though it confused me. I guess is my verdict on it, where it's so cool to be able to do this tech, and I think we're getting maybe a little bit lost in the details and taking a step back to appreciate that there are still proteins that are being recovered from dinosaurs. Like
yeah.
that on its
eggshell, and like not even super well cared for dinosaur, just like fragments from a rock shop. You can still get this stuff out of it. That if this holds up, that's going to be a big deal, and this could be a, I don't know. It's tricky, like trying to be able to find stuff to do that's scientifically worthwhile with eggshell, with just like fragments of eggshell that you find at a site. This could open up some doors like that. Like it could be that you go to a site, you find some eggshell, and suddenly you can tell something really cool just from a few fragments of dinosaur egg. And it's exciting to think about, and I really hope that it trends in that direction.
So, I guess because of how it was stored, do you think it's one of those things where if you were to find some in the field, you wouldn't have to do like that crazy preservation like we talked about with if you were looking for the DNA in the bone? Is it you could maybe put it in a box, then move on?
Yeah.
Like it almost has that like
the thing is,
ease vibe.
We usually we find eggshell on the surface usually. Like it's very rare that you'll be digging in a quarry and then you'll find eggshell. I've never done that in my experience. I've only ever found eggshell on the surface, which means it's already been exposed to the elements and everything. I guess if you found an intact egg and wanted to take a plug out of it or something, you could do that. But yeah, most of the time you find eggshell on the surface and it's already been subjected to sun and rain and everything else. Yeah.
So that it's even more impressive that we're able to recover anything from it, given what we talked about with DNA earlier.
Absolutely.
Yeah.
Yeah.
It's
I
guess it's surprising.
It is. It really is. And part of me also just wanted to go, oh shoot, if some of these fossils are, these are bits of eggshell from a rock shop, from the commercial fossil trade, like, I instantly have an instinct to kind of recoil at that and be like, if we don't know the provenance of this, if we don't know exactly when it's from, why are we even bothering to study it? If we can't say it comes from this outcrop in this part of the world with this age of rock, then why are we even bothering?
So, I guess, correct me if I'm wrong, one takeaway that I had was this only works up to a certain point in time. Because it measures, right, that 50-50 ratio of the amino acids. Right. And then if you go further, like once you hit a certain time, it doesn't tell you any older, and that's like how you tell against contamination. So is it something where it would be great if we could know exactly where it is and where it was from, but in terms of the testing for contamination, like you're kind of set on that with a particular date?
I don't even. Yeah, you gleaned more out of this than I did.
That was because I think what they were arguing was that to get to this 50-50 state of the both forms of the amino acid takes time, and at a certain point in time it hits that 50-50 mark, and then that's it. Like it stays at this 50-50.
When you say it hits a certain amount of time, do you mean during diagenesis, while it's being fossilized, or while it was there in the ground, or after it's been excavated? Where does the clock start?
That's a great question. So it starts whenever something dies.
Okay. Yeah.
And then it just immediately you start transitioning, because like the only thing that biological life can utilize is this one form of an amino acid. So if you're alive, you won't find any of the other form of the amino acid in your body because your body can't process it. But then there's this transition over time between those two forms, because there's nothing acting on misfolded proteins or proteins that shouldn't be there, because all your repair machinery is just gone because you're dead. And so it doesn't seem like, to me in this, it was right when the sample was taken out of the ground. Like you said, the eggshells are just exposed. So if you measure and look for these amino acids and you see that 50-50 ratio, then it's not contaminated, because and it's hit that point in time where it's become that 50-50, and so you can't use it to date anything. It's more of a quality check to make sure you haven't contaminated it. Because probably if there's like a peak in these dinosaurs, let's say your Syrian has a peak, like that shouldn't even be there. But then if there was a peak, then it's not that 50-50 anymore, and something's like really wrong with your specimen.
Yeah, I hear what you mean. Yeah.
But
yeah, for me, I think it was more like, oh, sorry, go ahead before I make my point.
Just to me, that I think the best way of having this data would be like a combo of what you said, like knowing the location, the strata, like not just. I took this more of as a proof of concept paper, and not necessarily the ideal way of doing the science.
Yeah, I guess in a way, sometimes there's a tendency toward a slight bit of snootiness on my part, where it's like, well, if we don't know where these fossils are from, then commercial specimens, like, blah. But like one of the authors said in the press release, like that was one of the reasons why they were able to do this kind of destructive sampling, is because these are just specimens that were basically stolen from Argentina, and we don't know where they're from, and they're not part of museum collections or anything, so that they could destructively sample them and just kind of do this proof of concept. And that's cool, yeah.
Do you think it's a worthwhile bit of data?
I don't know. I don't know what you'd really be able to. You're saying, like, aside from the gee whiz aspect of this, like, oh,
yeah,
that's cool,
like what would, like, in your view of paleontology as a field, what would this add?
I don't know enough about biochemistry to be able to say confidently, like, yes, this could tell us something cool about these animals, or no, it's, you know, it sounds cool, but it doesn't actually enhance our understanding of their biology. I really don't know. I mean, if you had a wide sampling of different kinds of dinosaur eggshell, and then you noticed some sort of a pattern across them that was consistent taxonomically, it's like, oh, well, ornithischian dinosaurs, their eggshell always has this kind of signature, versus Saurischian dinosaurs, it always has this kind of signature. That could be useful, I guess. But I don't know if that would be possible, and I don't know if we would expect to find, you know, with how weird eggs are and how much variation they can have, I don't know if we would expect to find, like, a nice cut and dry difference between the eggs of different clades of dinosaurs. Yeah.
And that's a little bit answered here, right? Just in this figure, too, of, like, here's different dinosaurs, right? Mm-hmm. The scales are different, unfortunately, which makes it difficult to look between the panels, because, like, one scale is to 0.4, the other is just to 0.6, but if you just look at, like, the global picture, it does seem like they are quite similar in terms of the amino acid composition that you're able to pull out, which, again, is not what I would predict, like, based on all we've talked about differences in the eggs.
Uh-huh.
And I guess the reason I was wondering, like, what you saw as, like, the long-term value to this is primarily because there was not a future direction section of the discussion. You know, of almost like why do I care beyond that it's really cool, and yes, like, I am from an intellectual perspective excited about it, but what can we get from this? Like, because it's not suggesting we can get anything outside of eggs and eggshells. Like, it's not like I'm doing proteomics on a dinosaur.
Right, yeah. It is kind of funny how they don't really talk about that in the conclusions. These results bolster excitement for the potential for eggshell calcite to aid in the study of ancient organic degradation. So it's more of a, not what can this teach us about dinosaurs, but what can this teach us about diagenesis and fossilization, and whether we'll be able to find biomolecules in certain fossils in the future.
So is it
Yeah.
then just a way to date things? Like, by looking at this ratio, as long as they're younger than that defined length of time, You look
But I
at
don't know if that defined length of time is actually going to be a useful parameter there. We can already date things like fossil eggshell. We can't date them directly, to my knowledge, yet, but we've got radiometric dating in goodness, it's getting late. Words are slipping my mind now. Zircons that we can find in the rocks. So we've got other ways of doing that. I don't imagine this would be a particularly precise way to actually tell how old these eggshells are or anything. But I don't know. Maybe this is just the very beginning of a process where it'll get more and more precise. I don't know. Yeah.
yeah.
I don't imagine it would be, but I don't know.
I'd like to imagine that it will lead to a lot more. I just honestly don't know.
Yeah.
What that lot more is going to look like, or it's going to end up being.
Mm-hmm.
Which I'm frustrated by, because I think this is really cool. I just, and I'm like, am I just being too thick-headed that I can't see like the immediate long-term benefit of it? That's why I was asking. I'm like, what do you think this is going to be used for? Like, it's cool, but like, it's like in an eggshell, and I wonder how much variance there's going to be between eggshells, or can you even get the resolution to see the variance, like we were saying? And I don't know. Still really, really cool, and I'm very excited that we were able to talk about this. But yeah, I felt like it should have been higher when reading the abstract in terms of the publication location, and then when it was like, okay, there's all these limitations, I kind of understood why it was just in this journal. And I feel bad for the authors, because I feel like this probably wasn't easy to do.
Oh yeah, and they probably shopped it around to a bunch of different journals and had to kind of settle for this one.
Yeah.
I've never actually heard of this journal before. Geochimica et Cosmochimica Acta. Yeah. I never heard of it before.
I was banking on you having heard of it, because I'm like, I have no idea.
No, never. I actually kind of wondered. There are a bunch of different journals that have Acta in the title like that, and I'm not Acta Geologica Polonica, Acta Geological Sinica, for like it's based in Poland versus based in China. I wonder what that suffix or that I don't know what that word actually means. I've never really thought about that until right now.
It is the plural of actum, a Latin word referring to official papers or documents.
Gotcha. Okay, that makes a lot of sense. So it's almost like saying proceedings or something like that.
Yeah, it just seems almost like a bit of a shtick.
Yeah, that makes sense. But yeah. Super long author list for this as well. Like, this was... is there an author contributions part? Because I'm guessing that's going to be pretty long as well.
That's a great question.
I'm not seeing one here, actually.
I didn't see one either. No. So, I guess, is this a new thing? So, I know it's relatively new in the biological sciences for, like, at the very end, you have the acknowledgments, but then you also have author contributions. Like you were saying, that's pretty new in the sciences, and I think it's a way to track, like, what exactly people did and make sure there's, like, enough of a credibility. But also, like, if something goes wrong, then you know who to blame.
Yep, yeah.
But yeah, there's none in this one. I don't see any, like...
It doesn't seem like it's super standard across journals, but I've seen it more and more recently.
Okay.
Yeah.
Yeah, the author location was also quite massive. Like, Chicago, the UK, Italy, Copenhagen, Denmark, Cambridge, Hong Kong.
You are
like
wholesome as
Madrid.
Yay!
Evansville, Illinois, Arizona. Like it's a huge location list, and I wonder how much of it is just like giving the sample, because
it could be one of those situations in which someone gets an authorship on the paper just for having provided one of the specimens or something. That does happen.
Yeah, because so just I know you have to go soon, Danny. Just so like some of the, I think the most Herculean part of this is doing this part here, the actual mass spectrometry. So building the reference database so that if someone did this again, they'd have something to plot against. I think, and like having a program that they could use, like that's the hardest part, because each time there's one of these like new things in a mass spectrometer, like we said, you have to develop your very own pipeline to analyze the data, because there is no like downloadable program. Like in sequencing, you can go into the R package, download ggplot and cufflinks and whatnot, that's like set programs, and they'll analyze the data with, assumed parameters, but it works on any RNA sequencing data set, and there's not equivalents to that in mass spectrometry. And so like this is the hardest part.
There's no equivalent to that in the sense that it hasn't been put together yet, or there's no equivalent because just that's how these analyses work, where you're starting from scratch every time.
Someone just hasn't done it to put it together. Like there is, I forget her last name. I just know her as Lindsay. She was a colleague of mine. I forget. It starts with her last name. Starts Pino, Lindsay Pino. She was working on creating a set of lectures and resources to standardize how we even do this, because it's like different labs also do it slightly differently. There's no one way of doing it. There's no company that put out a software to analyze this, and I think collectively that's led to, and also like the machines are $1 million, right? So someone in chat actually pointed out earlier when we were like, oh, how can paleontologists use this? It was like, with what funds? Right, like you have to have that collaborator. The service contracts, Danny, for one mass spectrometer a year is $50K. The service contract is $50K.
That could be 5 summers of digging out in the field for dinosaurs.
Yeah, so it's because of the price, it's not standardized. They have to redo the protocol every time, and because of that redo, you're left with this huge difficulty of comparing and contrasting. And Wombat pointed out, calibration's also key. Like, it's not just a plug and play kind of machine like an RNA sequencer, but you have to clean out stuff, and like the metal piping has to be switched out all the time. Like, there's so much nuance to one of these machines, in spite of the fact of how expensive it is. So it's it's just really hard to be able to run it. That's why I think this is the coolest part of the paper, is that someone figured out how to do this.
Yeah. That's funny. I was in reading through this paper, I was excited to hear your thoughts on it and kind of have you explain some of these aspects that I did not understand. And
did I do it justice? Did that make any sense?
It did. No, it did. But it kind of. I thought maybe there were certain parts of it that I just wasn't grasping, or it turns out that it's also just extraordinarily complicated. So my initial instinct to go, wow, this is complex and I don't understand it. I realize now, not only do I not understand it, but that's because it is incredibly complicated, much more so than I even initially realized. And it kind of makes this work, I'm not going to say it makes it more interesting to me personally, but I respect it more than I did originally.
Yeah, like
based on your explanations.
There's very few people in the world that are able to run these machines well, and like, it's, from my experience also, it's not the lab head. Like, there was this guy at Penn. He was like the premier mass spectrometrist on the East Coast. Which, if you can imagine, there's something like that. That's a weird thing to have as a title anyway. He had 6 of these machines, constantly running, but he had 2 technicians trained full time just to troubleshoot the machines and run samples. So it's not even something that you yourself, as even a molecular scientist or anything else, like, if you're not trained on this thing, you hand it off to someone. Like, that's how specialized a machine it is, and like it's not ever straightforward to learn and bring into anyone's
How
life.
many equivalent mass spec machines do you think exist in the world today? How many of these are there?
So to give it, I guess, perspective, I think there's several thousand sequencers minimum.
Okay.
There'd be a few hundred mass spectrometers. Is the scales that we talk about the differences, and again, it's because it's a great idea, but they're like to do the experiment, like even on living things, right? But it's expensive to house, it's expensive to maintain, and then it's honestly cheaper just to outsource it. So send the sample to a university or a company that has the machine, tell them what you want, and then they'll send it back. Again, the catch is troubleshooting. Who's going to do it?
So, for perspective here, compare that to like an SEM, like a scanning electron microscope. How many of those do you think exist? Is it more or fewer than mass spec machines?
Far more.
More SEMs?
Yeah, I think it's, historically also you're looking at something like a more ancient tech. So like ancient, right? It's not that old. But relatively speaking, and it's a way, like a standard metric for a lot of labs to use and look at, even though it's expensive.
Sure.
Same thing with, like, I guess confocal microscopes also run you a cool million, and those are like any university has it now. Like a confocal microscope is standard. It seems like most universities have an SEM as just standard. Like, and same with like an RNA sequencer or genome sequencer. Like a university will have that machine. Is like pretty, like, I'm sure Berkeley has got like 4, right? Like I would assume that the California school system, because they're really, really like, not only are they like high level in terms of the research, but it's like they're the leading schools in the state, and the state prides itself on its education, right?
That's
so
a prestige thing. They're going to make sure that they have that for sure.
That too, yeah.
Yeah.
And Nanula says I did my master's thesis on mass spec. Nice, yeah. We're dissecting away some mass spec here.
Huh.
And also, there's 2 kinds of mass spec machines. There is the LCMS that's used here, but then there's GCMS, so gas chromatography, mass spectrometry, and that's to look at hydrocarbons and odorants. And so, both have the issue of price, of maintenance, the database analysis, all of that. Like, both fall into having the same issues. And actually, but the data output is the same. It's all these peaks, and the intensity of the peaks tells you information about it. So it's like you read the data the same way.
Interesting. Well, it's fun to have these conversations because it's a reminder of how little I know about certain areas. It refreshes my level of respect for researchers in other fields. So, thank you for that, Belinda.
Thank you, Danny. I mean, it's the same for me. I'm just like, like, it's so cool to be like, oh, it's in dinosaurs. Like, I have no idea what this would be used for. Like, the logistics too, and like, I love these crossovers because it gives us a whole bigger appreciation for these papers.
Definitely, yeah, and I would have understood these far less without being able to talk to you about them. So, this is neat stuff, and I'm looking forward to the next one of these that we can do.
Yeah. Well, are we on for next week?
I don't see why not. Yeah.
Perfect.
I'll be on the lookout for some new papers, and I'm looking forward to it.
Yes, sir. Thank you, Danny. Is there anything upcoming in the world of paleontologizing the party people should know about?
Oh boy. Tomorrow I'm starting my stream early and then ending it early because I've got a meeting that I got to go to in the afternoon. But it's Thursday, Birthday tomorrow. And then Friday, we're going to be talking about the Bone Wars yet again. This time in kind of the Wild West days of dinosaur paleontology here in the U.S. After the Civil War, these 2 guys had this huge feud, which resulted in the naming of like over 100 different dinosaur genera. Probably about 20 of which are valid today. But it's the anniversary of the death of Edward Drinker Cope, and we'll be telling the really interesting story about how his skull was preserved in the Academy of Natural Sciences in Philadelphia, and then it went on a road trip back in the 1990s, and all these paleontologists got to hold it. They got to meet Edward Drinker Cope, this legendary figure from Dinosaur. We'll be talking all about that. It's going to be a good time.
And there's a really, really good book that you had suggested years ago. I bought from the car. We were driving over the bridge from Pennsylvania to New Jersey.
Was it this one?
No, not that one. It is.
Oh, the Dinosaurs of New Jersey one?
It's called Bone Wars.
Oh, yeah, by Tom Rhea. Yeah.
That's it.
What's funny is it's not totally about the Bone Wars. It's about, it's mostly about Diplodocus and Andrew Carnegie and all that. But yeah. It's from just after the Bone Wars, but that's the title, yeah.
I remember you had suggested this. We were driving on the interstate home, listening to the stream in the car, and Lita was like, "Get that book!" So we all of the
it's
dino.
It's a great book, isn't it? Yeah.
It really is.
I reread it. I reread it a couple summers ago, and I enjoyed it a lot. It's pretty cool. It's about, if anybody's watching from the UK, you're probably familiar with Dippy the Diplodocus. That's the story of Dippy, is chronicled in Bone Wars. But this is different. This is the skull of Cope here, maybe. If I can.
Oh, it is. They're just posing with it.
That's Jim Kirkland, the state paleontologist of Utah, who I work with during the summer. There he is with the skull of Edward Drinker. It's a wild tale, but we'll be talking about that. We'll be talking about the Bone Wars. We'll be talking about all kinds of stuff on Friday. It'll be a good time.
Nice. And I do want to plug, you've got the field excursions coming up soon. Y'all, there is the Amazon wish list for Danny to help with the field excursions.
Nearly depleted now. It's amazing, like the number of items that everybody's purchased. We've got hardly anything left, but
there's a couple of stuff though, chat. Y'all can get in and help. And this isn't just a one-off.
Oh yeah.
I think I love that you've made that very clear. These will be used for multiple trips coming. Yeah. So I've pinned it to the top of the chat. And y'all, there's no information. Danny doesn't know your address. You don't know his address. It just goes straight to...
It doesn't even go to me, actually. It goes to Ethan in Utah. And, yeah. But, that being said, if anybody purchases any items off of here, send an email to dinosaurcampaign@gmail.com. I'll type it into the chat. Letting me and Ethan know what item you purchased so we can put a little label on it with your username so that we can show it off while we're streaming from the field in Wyoming for the entire month of June. We're going to be out there digging dinosaurs, and you'll see some of these items in use. And, yeah.
And I go ahead and...
Thank you for the shout-out, Belinda.
Always, yeah. I went ahead and put it on our Discord, too, y'all, with the email address associated with it. So if y'all wanted to purchase something and be like, hey, that was me, it's in our mention on Discord, or mention on stream tab on the Discord page as well. So please, please, please support Danny, y'all. He, again, science on Twitch would not be possible without this man. He is the pioneer and the reason why we have this category. Support him on the Amazon. Go follow him on the Twitch. Go sub to him. He does this full-time, bringing us education on the daily. Like, freaking hero, Danny.
I do what I can. Again, Belen, what you're doing here is incredible. This incredibly welcoming community that you fostered here is truly impressive. And how close are you now to Partner Plus?
We're on the final...
You've got 2 of 3 months done so far, right?
Yep, 2 of 3 are done. We're on the final month.
Nice.
As of before stream today, we were at 123 out of 300 for the month.
Very nice. Okay. And it's only the 10th.
Yep.
You've still got like 20 more days left in this month. Beautiful. And
making that headway.
That goes well, I think.
We did turn in our the new ambassador application a few weeks ago. Because
very nice.
You know, we turned in the original one, and then we got I saw a tweet like 2 months later. There's no official page. They're like, oh yeah, we're redoing this. Everyone needs to resubmit. Good thing I'm on Twitter, right? And we saw like so there's a bunch more questions now, and so we rewrote the answers. We filled it out, and we found that there was a global document text limit. There wasn't one written.
Oh.
But
Uh-huh.
like getting the next to last question, like it stopped taking the answer. I was like, huh. And so then we had to go and edit it. But that's
Go back.
into.
Oh man.
Yeah. So
Yeah.
I was talking with Lita. I'm like, oh, if we get those two, I am not even putting in my 2 weeks notice. I'm taking all my vacation time, and during that time, I'll put in my 2 weeks.
Nice.
Bye.
Yeah.
Yeah, chasing and I wouldn't be able to have this dream and this like goal without you being an inspiration here on Twitch. And I hope many other scientists will see the same thing and be like, this guy has a love for science. He's bringing it to people who are then excited about it and paving the way for others. And so, Danny, thank you for everything that you do.
Thank you, Bullet. I should say the same exact thing for you. And I'm so excited for how your channel has grown, and I'm looking forward to a lot of science collaboration in the near future with you and with some other folks on Twitch. So, yeah, good stuff. Thanks for providing such a wonderful platform for this crossover, Bullet, and I'll see you later.
Yes, sir. I'll see you tomorrow, Danny.
Take care.
A
Bye.
good one. Bye. All right, y'all. That was Professor Danny Anduza. Make sure you go follow him. We gave him the Fancy Pants shout out. We will continue to do so, as we always do. He's one of our best friends on the platform. And again, I cannot... It's not just because he's here, y'all, that I say this. He truly, without him being here, I don't think the category would have ever taken off. Or it'd be like a viable means by which people can do outreach. So, y'all, please go support our friend Danny. Again, only because of him that we are now having like this opportunity to have education free on the platform for as many people as we do. So big thank you again to